Okay, so we're going to get started with our next session of speakers. Can I please ask everybody to get into your seats? Okay, for our next speaker, we are going to start this session of bioinformatics trainings, networks, and groups in the region and abroad with the Welcome Connecting Science. We have online Alice Matimba. She is the head of training and global capacity at the Welcome Connecting Science. So Alice, can you hear us?
Yes, I can. Can you hear me? Hello? Hello? Hello, can you hear me? Can you hear me?
Yes, thank you.
Shall I
You can
speak
get
again?
started now.
Okay, can you hear me clearly? Hello? Okay, I'm assuming you can hear me. Hi, hello everyone. Okay, I'm assuming that means you can hear me. Hello everyone. So my name is Alice Matimba. I'm going to be speaking to you on behalf of the Welcome Connecting Science Learning and Training team. I'm unfortunately cannot be there in person, but I'm with you in spirit. And I just want to mention, acknowledge that the work that I'm going to present, or the materials of my presentations, are a combination of work that has come from many people. And so it's not just my work, but it's work that has come from my team and other people also externally from our team. So I'm going to tell you a little bit about the program that we run, the training program that we run at Welcome Connecting Science, and particularly focusing on the global strategies and the community development strategies that we have employed to reach many people globally. So Welcome Connecting Science, for those of you who don't know what Welcome Connecting Science is, we are based at the Welcome Genome Campus alongside the Welcome Sanger Institute, and we are developing training and also running a lot of conferences that are available to people globally. As part of the Welcome Connecting Science, we also have the Engagement and Society, where there's work around public engagement and also training of other audiences that are not necessarily professionals. So learning and training is really focusing on training research professionals and healthcare professionals. So Welcome Connecting Science Learning and Training program is developing. It has a program that is covering various major themes from human health, human environment interactions, and also emerging techniques and tools. Our program is based, although we are based here at the Genome Campus, we have residential courses that are running here. We also have courses or training that is running across all over the world, in Africa and Asia and in Latin America. We also have a suite of conferences that are running also every year, based here at the Welcome Genome Campus. and also an online MOOCs program. And I'll tell you a little bit more about that shortly. So we really want to focus on training that's covering basic research, cutting edge medicine, and also how genomics is applied in healthcare. So started about 35 years ago. To date, we have run over 400 courses and conferences. And we are currently, over the last 5 years, we have reached over 200,000 people. And this is through our conferences, our hands-on training, lab and informatics courses, and also our online courses or MOOCs. So we apply various strategies on our training program. And obviously the main goal is really to build skills and capacity through short courses, where we apply different formats of training. We are also interested in making sure that our participants that attend our program are provided with tools that help them, that empower them through communities of practice and mentorship. And also we are, as we work with a very global stakeholder community, we are working with scientists to understand the needs and therefore try to make sure that our is meeting the target or the needs of the target audiences. And through all this, we do make sure that we are embedding equity, diversity, and inclusion, and also sustainability strategies. And some of these I will talk about shortly. So I just want to highlight that a major part of our program has focused a lot on pathogen genomics and capacity building. And this is through short courses. We've used various formats, and this has enabled us to really train a lot of people, but also to experiment on different ways that we can really build capacity, not only here in the UK, but also globally. So our program has over 30 courses and workshops that are delivered in face-to-face format, virtually, and also in geographically distributed classroom formats. A lot of our materials, particularly the bioinformatics materials, are accessible via GitHub. We also have over 20 MOOCs that have been developed over the last couple of years that are spanning, content from bacterial genomics, viral genomics, and also just basic bioinformatics tools that are important for example, for people that don't have much experience in very basic skills such as command line. So we have 2 courses that have been developed that are covering bioinformatics for biologists, which are very popular. And then, of course, we also cover other areas such as genomics for educators, genomics scenarios in primary care, what is genetic counseling. So we do have quite a wide range of courses and workshops that we have developed and that are currently running. So here I'm just showing you a map of what our content covers. So like I mentioned, we do have a very strong pathogen genomics component. And here I'm just showing you along the sort of like end-to-end pipeline of how genomics is applied from and all the way up until how it's applied both for public health and also for clinical care. So we have courses that are obviously focusing on techniques that allow people to train in from sample collection all the way to applying the information for genomic applications. And then, of course, we do have a very strong component that is covering just the bioinformatic analysis steps as well. To enhance the skills and capacity development, we have developed a different formats and different ways that we want to be able to increase the capacity of genomics through the trainer program. So we have different formats that we've developed, from in-person to online to sort of, having more, integrated forms of train the trainer programs. And we've mapped these across the different types of skills or basic competencies that are covering everything from just how to train in terms of basic analysis, all the way up to how to train in terms of communication. We have also tried to put this into some pipelines that are, again, available upon request, and also some of it is available online, freely available online. So I just want to cover here a couple of examples of how we've started to really support communities of practice. And this is through working with participants that have been on our programs to try and keep some of those communities going, to try and keep people that are interested in continuing to develop skills, in continuing to develop their own professional skills going. So we have developed an AMR network that is currently have more than 400 members, and interestingly, from all over the world, for people that are interested in AMR genomics focused on Africa. We have also been working with a recent cohort of participants from a single-cell genomics course in Latin America. And this community is going to be focusing on mentorship, but ensuring that we are embedding other skills like how to write grants, how to write, how to, analyze data, and also how to publish their work. And there are other communities that we are also developing, and we are learning from the process of how to actually make these communities, how to strengthen these communities and to keep them supported and focused. And we're quite happy to discuss further some of our experiences that we've had in developing these communities. So I really want to sort of like bring together all these different strategies that we have now developed expertise in, particularly in the organizing of training at scale, and organizing and developing of genomics training and bioinformatics training at scale. We have also obviously developed really just some really strong infrastructure and platforms that allow us to develop and deliver training at high levels. And we have quite an expansive global network of trainers and partners that enable us to be able to do this. So during the pandemic, we have applied all these tools. So the way that we are running, sort of virtual courses, or, the distributed classrooms, or developing communities. So we brought all these strategies together to as a way to respond to the pandemic and develop training for global audiences. So we used all these different methods. We used the online MOOCs, as I mentioned earlier on. So this is the courses that are usually running on that we run on FutureLearn, but also allow people to sort of interact and are facilitated asynchronously. We also ran virtual courses and also ran distributed classrooms, and later on we did have some community network activities as well. So this is just a flash summary of the achievements of some of these activities. So here you're looking at the results of the MOOC program. This was just focused on the SARS-CoV-2 training. And here you can see that our approaches actually allowed us to collaborate with over 120 experts from 27 countries, and reaching over 15,000 learners in 140 countries. Then we also worked to develop course which actually had a little bit more hands-on, to reach in a geographically distributed classroom model. We worked with hosts from about 27 countries, and were able to train over 300 participants in just basic bioinformatics. We then also ran some virtual courses, and these are a little bit more intensive week-long courses that allowed participants to sort of like have that end-to-end training in viral bioinformatics. And this was run for participants based in Latin America and also in Asia. And finally, we were interested in actually sort of like bringing all this together and develop a a program that allows people who are already in institutions where they are starting to develop some genomics capacity to workshop around their needs, and also to think about what type of processes that they would need to develop if they needed to establish genomics capacity in their own, in the context of their own institutions. So we ran a capacity building workshop where participants were able to really come together from Africa and put together almost like an action plan of how they were going to implement genomics in their own institutions. So we have put together all these resources online, so you can access them via our GitHub repository, and I'm happy to share the links after this presentation if you want more information. So all the online courses that I've mentioned, about 5 online courses from online MOOCs have been deposited on GitHub. We also have the virtual courses and also the capacity building workshop. So these are some of the highlights from this program, where we have now passed on these toolkits to people that are interested in developing their own training in their own local environments. So really translating these models or these strategies for use at a local level. So last week we had a course, we had 2 of our developers in Brazil running one of the courses, which was now translated into Portuguese, being run in Brazil. And this was run for 40 participants. And we're currently working with a team that's going to be also translating some of the materials that we've developed on this project to Spanish, and it will be run in Colombia in early next year. We've also now started trying to put together, a community where we're inviting all the people that have been involved in this project to come and share knowledge, but also to come up with activities that allow this community to continue to share and to share with each other and learn from each other. So in summary, this project is a result of all the, sort of like our expertise and infrastructure that we've built on the Wellcome Connected Science Learning and Training Program. We were able to develop MOOCs, we're able to develop train the trainer and capacity building training. We've been able to put together some training resources and guidance that allow other people who want to develop their own training in their own environments to be able to do so. We have a global network, we have an online learning community, and also training in different formats. So we've developed 7 courses on the MOOC and virtual and distributed classroom formats. Actually, it's supposed to be 8 courses. And we also have reached over, many people from all over the world. And this is quite substantial over a period of 18 months. So in summary, we have used our dedicated expertise in training development and infrastructure, which allowed us actually to have it enabled a quick and coordinated response to bespoke training for a pandemic. We used a mixed format approach, facilitating this global network building so that anyone, regardless of location, experience, or resources, is able to access training. And this is obviously a way that we want to really advance our training without borders strategy. We have also evaluated and we have demonstrated that scientists or people that we've worked with are able to cascade their knowledge and skills to others. And this is really important for capacity building and also for developing sustainable and collaborative communities in pathogen genomics. So before I sign off, I just want to mention that on top of all these things that we're doing, we also have a podcast, and I'm very proud of this because I think that it is a podcast that everyone should listen to, especially if you're interested about mentorship and careers and professional skills development and skills. So please do tune into Your Digital Mentor podcast on any of your favorite podcast apps. So I just want to acknowledge the Welcome Connecting Science Learning and Training team, and of course, many of the global collaborators that we've worked with over the last couple of years to develop this big training, global community and training program. And I would like to say thank you, and feel free to contact us if you have any questions about our program. Thank you.
Thank you, Alice, very much. And thank you in particular. I know it's very late in the UK right now. So due to time, maybe we can have one question for Alice.
Thank you for a very nice presentation. One question is whether there's potential for synergy between the kinds of trainings that Welcome Connecting Science is doing with what EBI is doing in terms of capacity building, and what that might look like in the future.
Yes, indeed. And I guess that's why we are on this panel. So yes, of course. And I mean, we welcome having those discussions, and feel free to contact us at any time. We have worked with EBI actually on a lot of and quite a few occasions, but maybe it'll be good to look at this in a more sort of like serious manner. So yeah, please do get in touch. Thank you.
Okay. For our next speaker, we have Nicolas Palopoli. He's a computational biologist, adjunct researcher at the National Research Council CONICET in Argentina, and works at the National University of Quilmes in Buenos Aires. In 2010, he co-founded the RSG Argentina, affiliated to the ISCB Student Council. He has been secretary of the Argentina Association of Bioinformatics and Computational Biology since 2017. Nicolas is also co-executive director of Metadocencia, an online community of researchers and educators that build scientific and technical capacities through the co-creation of networks, learning spaces, and accessibility resources for Spanish-speaking communities. Thank you very much.
Gracias. I'm going to give this talk in Spanish. Muchas gracias, Rebeca, y al resto de las personas que hacen Cabana NET por el privilegio de compartir estos días acá. También gracias a organizadores y el equipo de interpretación simultánea por el esfuerzo para permitirme elegir dar esta charla en español. Empezamos. Bueno, vamos desde ahí. ¿Puedo pasar de aquí? ¿Está bien? Ok. Hay un espacio allí arriba si querés poner la imagen, que así no me tapa lo que tengo que decir. Gracias. Bueno, no me voy a detener mucho. Creo que no está funcionando. Bueno, ese soy yo, un poco ya me presentaron. Les voy a contar que además de mi trabajo como bioinformático, en bioinformática estructural de proteínas, también soy codirector de Metadocencia y hoy estoy representando más este rol, así que les voy a contar un poquito sobre eso, perdón. Pasamos, por favor. Metadocencia es una comunidad de base online que nació en marzo de 2020 como respuesta a las urgencias impuestas por la pandemia. Nosotros teníamos experiencia en educación presencial y remota, entonces decidimos ayudar a personas como ustedes, personas que debían enseñar de un día para el otro en forma online. Y haciéndolo a través de consejos concretos y basados en evidencia publicada para mejorar la experiencia de clase. Nacimos originalmente como un proyecto enteramente voluntario y con el tiempo obtuvimos apoyo de la Open Bioinformatics Foundation, una ONG estadounidense que se llama Code for Science and Society, y a partir de nuestro segundo año también de Chan Zuckerberg Initiative. Hoy nuestra misión, como dice allí en la pantalla, es construir capacidades científicas y técnicas de forma responsable y con una mirada local, a través de la co-creación de redes, espacios de aprendizaje y recursos accesibles, principalmente para comunidades hispanohablantes. Hoy no, pasamos por favor. Gracias. Hoy no vamos a tener tiempo de repasar algunos de esos consejos y revisar cuál es la evidencia detrás de ellos, pero les invito a que tomen parte de nuestros cursos. Lo que voy a hacer hoy es contarles un poco sobre metadocencia y cómo construimos soporte para que la ciencia y la educación abiertas sean más equitativas. Gracias. Todo este trabajo lo realizan personas como las que están en pantalla, que son el equipo de metadocencia, y otras personas que participan de nuestros cursos, personas como ustedes que están en distintas ciudades de Latinoamérica. Algunas de estas personas se dedican a investigación y docencia, tanto en el área de ciencias exactas y naturales como en otras disciplinas. Tenemos en el equipo programadores, diseñadores gráficos, abogados, politóloga, comunicadora social. Pero todas estas personas tienen en común que en general ninguna tuvo la oportunidad de recibir entrenamiento pedagógico antes de ponerse al frente de una clase. Entonces, con esa idea nos propusimos acercar algunas propuestas de herramientas prácticas para hacernos la vida más fácil a la hora de tener que hacer docencia. Pasamos, por favor, gracias. Así que trabajamos para crear esta comunidad interdisciplinaria de intercambio de conocimientos que llamamos metadocencia a partir de marzo de 2020. Y en esos primeros 3 años ofrecimos distintos talleres de educación no formal para enseñar a enseñar mejor, de alguna forma. Y hubo una necesidad que pudimos satisfacer porque en ese periodo completamos 81 talleres, son talleres cortos de 3 horas, destinados a más de 2.000 personas de 30 países que los tomaron ya como profesionales que hacían docencia en su mayoría, e incluso con hablando otros idiomas no español como primera lengua. Pero bueno, entre esas múltiples propuestas hay algunas que todavía mantenemos, como el taller presente, recursos para encuentros activos, donde cubrimos cómo hacer clases más activas con prácticas de mapas conceptuales, de evaluaciones formativas, de devoluciones permanentes, cómo construir un espacio de clase seguro y amigable. También tenemos cómo enseñar a programar online, que refuerza ideas de programación en vivo, programación entre pares, o Zoom accesible con lectores de pantalla para docentes y estudiantes que requieran ayuda en la accesibilidad en el caso que trabajen con personas con dificultades visuales. Así que todos estos cursos son cursos gratuitos con materiales abiertos y reutilizables que están disponibles en nuestra página con licencias CC BY. Pueden dirigirse en la presentación siempre al link que está ahí arriba o a la presentación misma con el link que está abajo a la derecha. Pasamos, por favor. Bueno, claro que las urgencias que habían nacido por la pandemia fueron cambiando y también cambiaron nuestras prioridades, por lo tanto, a lo largo del tiempo evolucionamos hacia trabajar en buenas prácticas de ciencia abierta. Nuestra visión actual es trabajar para que la producción, el intercambio y la aplicación de saberes científicos y técnicos sean globalmente equitativos, porque entendemos que de esa forma distintas personas se convertirán en participantes principales en la creación colectiva de conocimiento. Así que siguiendo la recomendación sobre la ciencia abierta de UNESCO de 2021, encontramos que hay 5 pilares claves para la ciencia abierta, que son el conocimiento científico abierto, las infraestructuras científicas abiertas, la comunicación de la ciencia, el compromiso abierto con actores sociales y el diálogo abierto con otros sistemas de conocimiento. Así que le voy a contar brevemente cómo abordamos estas ideas de la ciencia abierta en las distintas iniciativas colaborativas con las que nos vinculamos. Gracias. Primero les cuento sobre una iniciativa de la NASA, la Agencia Espacial Norteamericana de Estados Unidos, que está desarrollando un programa integral como parte del año de la ciencia abierta, que es este, con actividades para apoyar el avance de la ciencia hacia mayor apertura y que tiene el objetivo de formar 20.000 personas en ciencia y técnica abiertas. Así que en el contexto de esta iniciativa de la NASA, creamos 3 proyectos que fueron financiados este año por el programa TOPS, o Transformar a la Ciencia Abierta de NASA, y que están orientados a profesionales y educadores en ciencia y tecnología, muchos de ellos desde Latinoamérica. Y bueno, y como parte de estos 3 proyectos financiados por NASA en los que estamos involucrados para los próximos 3 años, tenemos un proyecto de formación de cohortes, de ofrecimiento de cohortes virtuales de formación en ciencia abierta, que se van a dar tanto en inglés como en español, y junto con OLS, Open Life Science, como compañeros en este proyecto. Estos cursos van a tener una duración de 6 semanas y la participación de alrededor de 50 personas en cada edición, y el objetivo es enseñar un currículum de ciencia abierta que fue desarrollado y validado por profesionales de distintas áreas y de todo el mundo. Además de esos 2 proyectos de cohortes virtuales en inglés y español, junto con 2i2c, vamos a hacer otro proyecto que está centrado en la enseñanza acerca del uso de datos abiertos de imágenes satelitales que comparte NASA, y la idea es que ese proyecto ayude a aprender cómo predecir sequías, inundaciones, incendios forestales a través del análisis de estos datos abiertos que tenemos acceso a través de los satélites de NASA. Pasamos, por favor. También somos parte integrante del proyecto Catalyst, que hoy Kate Herdwick de CCDI lo mencionó. Este proyecto, financiado por CZI, quien también financia metadocencia desde 2021, aparte de este proyecto, es un esfuerzo colaborativo entre 6 comunidades: metadocencia, CSCCI, OLS, Invest in Open Infrastructure, 2i2c y The Carpentries. El objetivo es ofrecer un servicio de infraestructura de cálculo en la nube, gratuito, con una duración de 1 año, con soporte, para que además se puedan correr en esa nube flujos de trabajo útiles en distintas disciplinas de biociencias, y que se puedan aprovechar por las comunidades que quieran acceder a eso para sus propios fines durante el tiempo de vigencia del proyecto. Así que si tienen el interés en utilizar estos recursos de nube, por favor charlémoslo, porque tenemos la posibilidad el año que viene quizás de invitarlos a participar. La idea es que sea un proyecto piloto que acompaña a las comunidades en el aprendizaje de cómo trabajar con este tipo de plataformas, que son abiertas e inclusivas desde la noción que las dirige. También el acceso mismo a esa infraestructura para que lo utilicen para sus medios y lo reutilicen, y enfoque también de formación de formadores hacia la enseñanza. Vamos a trabajar con comunidades de América Latina y África principalmente. Bueno, tenemos otras propuestas. Por ejemplo, a principios de año hicimos junto con Selene Fernández y María Bernardi un curso Train the Trainer. Fue la primera colaboración oficial entre Cabana NET y Metadocencia. Fue un curso presencial en Buenos Aires, basado sobre todo en material del curso Train the Trainer de EBI y de Carpentries, junto con material del libro Teaching Tech Together de Greg Wilson, que es un poco referencia en este tipo de educación en clases activas. Así que funcionó muy bien y ojalá vengan más colaboraciones similares. Pasamos. Como parte de nuestro trabajo colaborativo, estamos haciendo un mapeo de distintos eventos, financiadores y comunidades de ciencia abierta en Latinoamérica, y también haciendo el análisis de esos datos para poder comprender cómo las comunidades que trabajan en estos temas interactúan entre sí y qué rol juegan dentro de estas comunidades. Pasamos, por favor. Bueno, esto es un poco la extensión de lo anterior. Todas esas comunidades que colaboran con nosotros las estamos vinculando en nuestro panel de comunidades como una idea de incrementar la visibilidad de las distintas comunidades como Cabana con las que colaboramos, y a la vez, a partir de esta plataforma y de esta intención de colaborar, poder presentarnos a distintas iniciativas de subsidios, compartiendo, ya sabiendo que compartimos valores y objetivos generales. Por supuesto, están invitados a sumar a su comunidad del panel. Bueno, y un poco para cerrar, aprendiendo de lo que hicimos, quería reforzar algunas ideas y invitar a la reflexión. Aprendimos, como indica la evidencia, que sí, que trabajando en comunidad obtenemos mejores resultados y llegamos más lejos. La idea siempre que perdura es generar recursos pensados a partir de las necesidades locales, pero que puedan ser aprovechados por el resto del mundo, si son visibilizados, porque también creemos en que eso es una forma de combatir asimetrías de poder y privilegios cuando permitimos que el conocimiento que generamos llegue a otros lugares. La creación de capacidades la vemos como un punto importante en esto, porque apoya el desarrollo de la buena ciencia, aunque pueda no ser ciencia en sí. Algo que no es menor es que el trabajo voluntario no es sostenible, lo venimos experimentando en distintas comunidades y sabemos que necesitamos retribuir adecuadamente a las personas por su tiempo o aclarar las condiciones de trabajo cuando son diferentes a eso. Y por último, actuar por la diversidad, equidad e inclusión del sur global. Significa para nosotros tanto abrir espacios, es decir, crear nuevas oportunidades para otras personas, como hacer espacio en aquellos lugares donde estamos para que se puedan sumar otras voces. También es sumar a las personas en la toma de decisión, no solo como observadores, sino para que tengan voz y decisión. Y de esa manera también aumentar el valor de cada persona y su visibilidad en el contexto global. La última, bueno, ya para cerrar, tienen los links de la presentación. Quiero destacar que tenemos financiamiento de Code for Science and Society, que nos da patrocinio fiscal, Chan Zuckerberg Initiative, financiamiento hasta el próximo año, y de NASA para los próximos 3 años. Pueden contactarnos en redes sociales o unirse a nuestro Slack, y estoy dispuesto a contestar sus preguntas. Gracias, en inglés o en español. Contesto rápido.
Tremendo esfuerzo. Me pregunto si, supongamos que soy un profesor en alguna universidad donde tengo que hacerme cargo de un curso de posgrado, quizás mixto posgrado con algunos de pregrado en bioinformática, y no soy experto en todos los temas. ¿Hay recursos a los cuales yo puedo acudir que ustedes tengan disponibles como para facilitar la generación de ofrecer contenido o tutoriales o material y hacerlo parte de ese curso, y a la vez, si yo genero material en el contexto de mi curso, poderlo hacerlo disponible a través de la plataforma de ustedes?
Más o menos, lo que vas a aprender con nosotros es la forma de que cualquier contenido que esté ahí sea convertido en clases activas para tus clases. Es decir, cómo hacer que ese material sea más motivante y que logres una mayor eficiencia de aprendizaje gracias a que el material y tu clase así lo apoyen. Los materiales técnicos no los compartimos, no porque, digamos, es por diseño que no es así. Sí nos valemos cuando enseñamos con nuestra idea de cómo dar una clase. Por ejemplo, el material abierto de Carpentries. No sé si conoces de Carpentries, pero te invito a que lo busques. Ellos hacen material muy bueno y con colaboración de muchas personas, donde sí, por ejemplo, puedes aprender Python para datos de ecología o geociencias con R o cosas así. Y eso sí tienen unas guías bastante buenas, que en general son cursos contenidos en 3-4 horas. Y lo que nosotros vamos a enseñarte es la dinámica de clase para que esos materiales no sean simplemente texto que vos reproducís, sino, por ejemplo, hacer una clase con demostración de programación en vivo y apropiarte de los errores como una forma de aprendizaje. Creo que no se ocurre rápido, así que...
No, I just wondered whether there was any possibility of translating the materials into Portuguese as well.
We started doing that. We have a Brazilian person in our team. So our basic course is translated, the resources for active meetings. The rest, we decided to focus on Spanish because we mostly track people in Spanish-speaking countries. So that became our mission and vision to serve them. but for sure, if someone is willing to, we're happy. No. Gracias. Gracias.
Thank you very much. For our next talk, we have Lady Osorio. She's a native from Peru. She holds a bachelor's degree in genetics and biotechnology from Universidad Nacional Mayor de San Marcos, and a master's degree in cellular and molecular biology from the Universidad of Sao Paulo, where she is developing also her PhD degree. She also serves as the president of the RSG Peru, where she actively engages with the students in the fields of bioinformatics, fostering collaboration and knowledge sharing. She also plays an integral role as a member of the board at the Peruvian Society for Bioinformatics and Computational Biology.
Okay. Good afternoon to everyone. Thank you very much for the opportunity to be here. Muito obrigada pela oportunidade. Muitas graças pela oportunidade. Meu nome é Cleide, como meu nome é Cleide. Meu nome é Cleide. My name is Cleide Osorio. I'm going to present today this talk. I would like to introduce, well, yeah, my introduction is here. I am, oh, sorry. I don't know. Okay. This is my career. Now I am Peruvian. I am making my PhD degree in the Universidad de São Paulo and in the Laboratorio de Biología Celular y Molecular Trypanosomatidiosis, and founding member of the Peruvian Society for Computational Biology, Bioinformatics and Computational Biology, and RSG Peru president. And I would like to tell you about the focus of my research. I am working with FASTEN proteins. FASTEN protein is localized in the flagellum attachment zone of Trypanosoma brucei. This is a zone connecting connecting the flagellum and the cell body, and this protein has a pivotal role in the correct cleavable in the cytokinesis. It's very important as fast organizer and make a lot of interactions for the survival of these parasites. My lab work with a protein with this important zone, and FASTEN has been characterized firstly in my lab, but now I want to know about the structure to unrelieve their unknown functions using tools for disorder prediction. Disorder predictions, colloid predictions, and now we know that FASTEN has colloid domains. We focus in the central region because the central region has, in addition to low disorder, colloid motif. This motif is very important to make dimerizations in the colloid can form dimerizations of the, I don't know what happened. Well, can generate dimerization of the proteins. We identified the hydrophobic amino acids that can make these dimerizations of this protein. And why we started with the central region? Because this protein has mass molecular protein, and we start as like an initiative or a start point to know the structure of this protein. We start with this region. Using AlphaFold, right, to know about the structure. Okay, and now the next thing that I made is expression and purification of this protein to obtain the protein, and the next step is to resolve this protein with X-ray crystallography, and make a standardization of the protocol to intend to purify all the protein, all the fasting, and use the cryo-EM to resolve the structure. Yes, to resolve the structure. This is the next step of our work. For example, this structure that here is that a domain globular of the central region that we now the protein the purification of this part of fasting. Okay, now the second part of my presentation is talk about the Peruvian Society for Computational Biology and RSG Peru. This is the name of in Spanish. Sorry. This is the name in Spanish of the society, the first society in Peru, and the RSG Peru, Research Student Group Peru, and the logos. And serving Peruvian researchers in bioinformatics from 2019. We are serving Peruvians bioinformatics training and supporting bioinformatics in Peru and Peruvian informatics around the world. In 2018, we made a census to know the situation, the status of bioinformatics in Peru, in my country. And now that in the capital, Lima, we have a lot of people working in bioinformatics in contrast to the province, other regions in Peru. And we have a lot of Peruvians working in bioinformatics principally in two countries, USA and Brazil, and some others in other continents. But the provinces, other results of this census showed that the proficiency of level of proficiency of bioinformatics of Peruvian is high in all of people working in other countries. So due to the result of this census, our efforts were directed towards enhancing the collaboration between Peruvian bioinformatics and experts in other countries, Peruvians working in other countries, and supporting and helping Peruvians in working in Peru, delivering international knowledge. Courses without course or with minimal course, with the participation of and collaboration of people working in bioinformatics, not only from Peru, and raising funds to support these initiatives and help people to travel, to have training. And the society has established new connections. As the last directory of international affairs, now we have this connection, this collaboration with these societies. For example, in research collaboration with REABIO, the Red Inteligencia Artificial para Iberoamérica, with Soy Bio, and organizing events with the ISB Student Council and the ISB. And we promote the participation of people in all the events possible of the ISCB. For example, these events, chairs, people in the organizing committee from Peru. For us, is very, very, very important. And we reached over 25,000 people, and we organized these events, talks, focused in women's roundtable. Each year, we organize anniversary symposium. The first edition of the award, the first event about hours about bioinformatics is our event, Bioinfo Peru Awards, certificate training courses, Bioinformatics Journal Club, 2 or 3 journal club per year, and the Regional Student Group Peru as a strategy to join efforts of both institutions. Now, the Regional Student Group Peru is the Student Council of the society. This is some events of some of our events. For example, this is the anniversary symposium, curso internacional with Riabio about diversidad, equal of gender, introduction on Python, for example, just with women, and our first webinars. RSG Peru is focused in students, high school students, undergraduate students, and postgraduate students. And we want to consolidate the computational biology and bioinformatics in our country. And well, I'm here to talk to you about the RSG Peru initiative. We start in this way to impulse the students to join us. And we have regional nodes where principal objective is to centralize the bioinformatics with a training in other cities in addition to Lima, La Libertad, Arequipa, Cusco, Ayacucho, in people were never listened about bioinformatics. For example, we participate in this event of the Student Council Symposium. We have an article about it. A lot of people in the organizing committee. And this is some events focuses always in for a student, right? Pasantías, progression studies, this is a convocatoria, taller LaTeX, for example, and some meetings in person. And just to conclude, the objectives of RSG Peru always is active participation in social networks, organize events for education and development, and alliance with other RSG, for example, Brazil, and Chile, and Cabana, of course, too, and other societies. And just to conclude, and our future event is the first symposium of young Peruvian researchers in bioinformatics, mentorships, and the article of the month to recognize people working in bioinformatics. And I think this is the last, this is our, for more information, our website and our social networks. I think that this is the last, the next slide. We want to continue promote the knowledge exchange and networking, partner with industry, academy, and encourage always and promote diversity and inclusive in the community. And it's just everyone is invited to collaborate, sponsor partners to join us in our mission to improve the bioinformatics in my country. And contact me. Together we can shape the future of bioinformatics in Peru. Thank you very much. Muito obrigada. Muchas gracias.
We have one question.
I think you have so much, we have so much to learn from your regional approach within the country to getting out of Lima and into more remote regions. And I think that's something that you could perhaps work with the other RSGs to develop in other countries around Latin America. That would be fantastic.
I was going to ask that, are there any connection or activities between the different RSGs?
No, I understand.
Are there any activities or connections between the different RSGs from the different countries, like RSG Peru and RSG Brazil, RSG Colombia?
Activities and with countries, with my country. Yes, we participate always in symposiums. For example, Hispanohablante Symposium with RSG Peru, just speaking just in Spanish, for example, and always participate in these events. Okay. In this type of events, really. Of course, we would like to participate more and have more participation of Peruvian people in other countries to help Peruvian people in the start of their career and to consolidate bioinformatics in general.
Excellent. Thank you.
Okay, and for the last talk of this session, we have Raisa Maria de Melo. Raisa has a bachelor's in biomedicine by the Federal University of Pernambuco in Brazil, with an exchange period of the University of Toronto in Canada, and a master's in bioinformatics by the University of Sao Paulo in Brazil. She currently works in Toronto at a hospital for sick children as an assistant research technologist. Her main areas of research and interest are epigenetics, neurodevelopment, and psychobiology, biological networks, and recently search for SNVs. And she'll be talking to us about the ISCV RSG in Brazil. This is going to be a virtual talk. So Raisa, can you hear us? Raisa.
Hi, can you hear me now? Sorry about that. I guess now it's working. My microphone was with no volume. Okay, thank you. Okay, first, thank you for the invitation. I'm so sorry I could not be there. I'm gonna make a quick presentation about RSG Brazil. So let me just put full screen here. So hi everyone, my name is Raisa. I am the actual president from the RSG Brazil. I'm gonna give a little presentation about what is the RSG and how it works. So RSGs are groups from the International Society of Computational Biology. The Student Council just thought they could improve the knowledge in bioinformatics around the world, including students. So RSGs are student groups across the globe. So these are all the countries that have RSG, just like a colleague just presented now from RSG Peru. So this from the ISCB Student Council website, the current active RSGs. So the RSG Brazil was initiated for the first time in 2009. Then it stopped around 2011, 2012, and then it came back in 2015 and is still active until today. And every 2 years we change our board. So this is how this is distribution around Brazil. So one of the main goals of the RSG is to decentralize the bioinformatics, because it used to be restricted more to the southeast of Brazil, like São Paulo, and the biggest city that we have. So the RSG came to kind of try to bring bioinformatics to the distance around the country. So now we are around 20 people. We are with the intention to grow a lot from this year, next year. We have some plans, and we're gonna open a new calling for new members at the end of this year. So this is the actual board from 2023. So I'm the president. So we have also the vice president, Sophia, two secretaries, and then vice president, Sophia, two secretaries, the treasurer, Sophia, two secretaries, vice president, Sophia, two secretaries, vice president, Sophia, vice president, Sophia. Something happened with the audio. Okay, okay, came back. Okay. So we have a treasurer and we have a faculty advisor. Okay. The audio is coming back. Okay, now it's better. So, and the faculty advisor, which changed also every 2 years. So this is our active projects in the moment. We have 6 active projects. I'm gonna talk a little further about each one. So we have training and learning, we have a podcast, we have a league, which is a competition bioinformatics that we provide here in Brazil. We have social media in which we share different topics about bioinformatics. We have this bioinformatics outside of the bubble, bioinfo fora da bolha, to go like outside of the academic bubble. And we have symposiums in which we try our best to do one at least once a year. So we have these active projects, and this constitutes the RSG Brazil right now. The podcast is in Portuguese. We have the intention to maybe do some English episodes, but for now we have over 20 episodes in Portuguese, and it's accessible in any audio platform. You can look for RSG Brazil podcast, and you can hear episodes. Like you have episodes from all kinds, since work, job market, and also women, women bioinformatics and other topics. So we have the league, as I said, which is the competition that we promote every 2 years. So the league, Brazilian Bioinformatics League, LBB, it has 3 phases. The last one happened in the X meeting, which is the biggest computational biology symposium in Brazil, promoted by the Brazilian Computational Biology and Bioinformatics Association, the AB3C. And then we have our events there. And you can learn more about the LBB if you go to the Instagram or the website too. So we have social media. This is the QR code for our website. In our website from the RSG, you can find like all every contact, all the media, the social media that you can contact us, also about our projects and who is a part of our group, also who were in the past. So we have like Facebook, Twitter, Instagram, YouTube. So this project is bioinformatics outside of the bubble that we called, because as I said, we wanted to bring bioinformatics to like schools and to other places beyond the academic bubble. Because we've been noticed that it's very hard to explain to people outside of academic bubble what bioinformatics is. When I say that I did a master in bioinformatics, people that is not aware, like people that did high school, for example, they have no idea what I do. So the intention of this project is to bring bioinformatics in an easy, accessible language. Every people, everyone that could, even like high school level people. So we have 3 kind of posters in which we divided by colors. For now, we just the Instagram page, but we have the intention next year to go to schools and do some bioinformatics promoting and show the importance of bioinformatics in general to schools, in schools. So we also have training education in which we do special interest groups and courses. So we had like 1 course. We are now organizing our 2nd course. That's going to happen next week, actually, the 15th and 16th of November. It's in Portuguese, but it's about docking molecular dynamics. And if anyone wanted to participate, the registration is until November 13th. We also have symposiums, as I said, once a year. We already had 6 symposiums, and next week too, we're going to have a 7th symposium right after the courses. It's going to be on 17 and 18 of November. And the registration is also still open until tomorrow. If anyone want to participate, all of our symposiums are in English because we are an international group, even though our goal is to promote bioinformatics more in Brazil. This is just the past editions of our symposiums. Our 2 last symposiums were online. This year, we're also going to be online. But before that, we had presential face-to-face symposium during the X meeting that happened that I mentioned before. And the last one in person was Women in Bioinformatics. That happened in 2019, before the pandemics. This is just the registration for our last symposium, which was online. We have over 500 registration, people from all backgrounds that you can imagine, even high school. We also, the league had 2 publications. There are another paper from this year that was submitted with a written answer. So we have like one in a Brazilian magazine and one in BioArchive about the league and the competition in bioinformatics in Brazil. So yeah, this is basically it. So we have like some criteria to select members, so then we can keep the work, we can keep everybody engaged because it's a volunteer work. We look for these characteristics in our members. If you from Brazil or you are now in Brazil, you're interested in be part of RSG or even collaborate with us in another way, even though you're not a student, can contact us. If you have any questions, you will be here to answer. And once again, you can contact us also by social media. We have our email. This is our QR code for the website where you can find all the information and also information on our symposium that I just said that happening next week. And yes, that's it. Thank you very much for this opportunity to share this.
Thank you very much. We have time for a question or a couple of questions. If there's anybody here. Cass?
Thank you, Raisa. That was really inspirational, and I hope she's still there. We can see you. Oh yes, we can see you again now. I'd just like to emphasize the tremendous work that the Brazilian RSG and the other RSGs do for ISCB itself. You really are the lifeblood of the society, and it's amazing to see so much energy. And I really just want to congratulate you and the other LATAM RSGs for what you're adding to the society. We can take a lot of inspiration from it. Thank you.
Also, thank you very much. So we like to do this work. It's a good network, and actually, if I talk for myself, I feel very excited when I see bioinformatics more spread around the world and people understanding the importance of the work I chose for my life.
Thank you very much. Okay, thank you, Raisa. With this, we're finishing the session for today. Thanks to all the very nice talks and the speakers that we have today. We can give a round of applause for everybody.
And then, okay, so they are grinded, and then they water is added to the juice and to the fibers, so that fermentation can happen. And then the fermented juice is then distilled, and that gives rise to agave spirits such as tequila and mezcal. Okay, and so what is interesting is that these fermentations, especially in traditional distilleries, they are open fermentations. So what this means is that the microorganisms that perform these fermentations, that they come from their environment. Okay, so they offer a perfect opportunity to get to know the yeast that live in these different ecosystems in Mexico, very diverse ecosystems, and but also open the possibility of better understanding this process that culturally is very important for Mexico. Okay, so to characterize the yeasts that are living in this and they are responsible for these fermentations, we perform a very large sampling effort. So we went all over the country in the producing regions. Here in the map, you can see the regions are shown in different colors. And you can see that they are distributed all through Mexico. And as you can see in the picture here below, we went and sampled these fermentations and froze them to go to the laboratory and then isolate yeast and analyze this data. So to date, we have sampled more than 120 sites, and this within them, we have sampled 230 different fermentation tanks throughout the country. And we also sampled some plants, insects, and objects, because we are also interested in the ecology of these yeasts. So how they come, they get to the fermentation tanks. So and this allowed us to assemble a collection that we are calling the Yeast Mexican Culture Collection, and it contains around 10,000 different yeast isolates. So it's a very large collection of different species of yeast. Okay, so from these yeasts, we sequenced several of the genomes, and this is where the support from Cabana was essential. So the work from Abraham and Angela. These are 350 Saccharomyces, so the common yeast isolates, but we also have genomes from other yeasts. Okay, and so this the report of the collection should be coming out soon. So and we hope that this week or next week is going to be accepted. And Abraham, as I mentioned, and Angela are authors of this work. But we think that the most interesting part is still to come. So starting to analyze these genomes, the first thing that we can see is that the yeasts from Mexico they form a different group. So what you can see in this phylogeny, all the dots are strains that we recover from Mexico, and the phylogeny was reconstructed from the whole genomes. And you can see here and also on this principal components graph that the Mexican strains, they gathered together apart from strains from other regions of the world. Okay, so this was quite interesting. And then when we look in more details, what we see is that the population within Mexico is quite structured. So the strains, in terms of their genetic composition, they gather according to the region where they were collected. Okay, so there's a genetic structure. And so this, we hope to publish this also soon. And talking to Adrian, for example, it would be very interesting what the physiological consequences of these differences would be, by maybe performing metabolic analysis to see what the consequences of these differences are. Okay, and then the finish, I would like to tell you of a second finding that we found very interesting, and that is the presence of hybrids of different species in these fermentations. So what I'm showing you here is the way we analyze the first steps on the analysis of the genomes. So what we do, these are coverage graphs. So you can see here how many reads map to the different positions throughout the genome, but we don't map these reads to a single genome. So what we do is we map these reads to several genomes of different yeasts to be able to identify the yeast and to see exactly the composition. So most of the isolates, they are from the common yeast, Saccharomyces cerevisiae. We find some isolates that the reads belong to sister species, Saccharomyces paradoxus. But surprisingly, what we also found is that many isolates map to two different species. Okay, so this means that the species have crossed, interbred, and we have many hybrids in the population. Okay, so what we think is, and the proportion of hybrids is much higher than what has been reported in other places of the world. So, for example, natural hybrids have been only isolated in 5 different regions, and there are only 5 or counted number of hybrids around the world. And here, as you can see, we have several dozens of these hybrids. Okay, so here, just to show you the composition. So in blue, you see Saccharomyces cerevisiae strains, in red, Saccharomyces paradoxus, and in yellow you see the hybrids that I've been talking about. Okay, so and what is interesting is that the distribution of these hybrids is not uniform across the country. So you can see that in the south, these hybrids, we haven't been able to isolate these hybrids, while in the center and north of Mexico, these hybrids are much more common and than in the south. Okay, so we are again trying to understand what makes this environment and the region a hotspot for hybridization, and this is what our work is. So as I mentioned at the beginning, so the work that was enabled by the Cabana grant has transformed a lot. So now the main players are Alexander de Luna and Lucia Morales from UNAM and myself, and we established a consortium that is called the Yeast Genomes Mexico Consortiums, exactly to characterize the genomic diversity of yeast in Mexico. And there are many different collaborators and postdoctoral researchers, graduate students, technicians, and of course it is important to thank the producers that allow us to sample their fermentation tanks.
Right. So a bit of Latin American research of global importance. So any questions? Okay.
Oops.
Very quickly.
Thank you. That was a really lovely talk, Eugenio. I'm guessing that there's more in the ferments than yeast. And I'm wondering whether there's any opportunity to do metagenomics and find out what else is in there.
Yeah, so this is something that we did. So we took 2 types of samples. We took some samples with a glycerol. That means that the organisms stay alive, but we also took some samples directly and froze them. And so we are doing the microbiome analysis to see what the whole community is, and there's a lot of bacteria, of course, that are important for the fermentations.
Yeah, I'm sorry. I'm sure there's lots of questions. I have some too, but we have to move on because we've got quite a busy schedule. But I certainly would like to catch up with you and ask you a few questions later on. So thank you, Eugenio. So our next speaker is Federico Vignale. We're very proud of Federico. He was one of our first secondees, and he was a real ambassador for the Cabana project when he came to the UK. We had no idea when the first secondees came how successful the project was going to be. So we were really grateful for the first ones that came. So Federico studied at the University of Buenos Aires. I think he was supervised by Adrian. He was a Cabana secondee, and he has been looking at microbiolites in high Andean lakes. And this has proved to be a really important bit of research, which again has global significance coming from the region. So Federico, please come up.
Hello, everyone. Do you hear me? Okay. Is the presentation ready? Well, I would like to thank Cabana NET, of course, for the invitation, and tell you that today I'm going to present a research project, which was a collaboration between the University of Buenos Aires, the Astrobiology Center from Spain, the University of Marburg, and EMBL. And well, here we are. So this project is the study of microbialites in a particular place, which is Pozo Bravo. But does it work? Ah, perfect. So before jumping into the research, I would like to give you a quick introduction about this field, which is geomicrobiology, because I think that most of you are not familiar with this field. So you know that in South America, you have the Andes Mountains, which are divided in 3 regions. You have the Northern Andes, the Central Andes, and the Southern Andes. But our area of the study is the Central Andes, which is where the Andes are divided into the Western Cordillera and the Eastern Cordillera. The thing is that over the years, the erosion of these mountains and the deposition of the sediments between these 2 cordilleras form what is called the Altiplano and Puna Plateau. This plateau is the second highest and largest plateau in the world, and it consists of numerous endorheic basins, which are forming hypersaline lagoons. For those who don't know what are the endorheic basins, are basins where the water can't reach the ocean. So the water is concentrated between these 2 mountains, and due to the evaporation, the radiation, and then the evaporation of the water, all the salt is concentrated between the 2 mountains, and then you have the Altiplano and Puna Plateau with these hypersaline lakes. So here are some images of these lakes. As you can see, they are super diverse. They have different colors. You can find red, orange, yellow, also like the depth of these lakes are completely different. But the most important characteristic of these lakes is that they harbor microbial ecosystems. Which type of microbial ecosystems? Well, first you have biofilms attached to rocks or on the surface of the water. Then you can find what are called microbial mats, which are stratified biofilms. And biofilms and microbial mats in some occasions have the capacity to precipitate minerals and form what are called microbialites. Microbialites are the first evidence of life on Earth. And then you have endovaporites, where are evaporites colonized by microorganisms, where the minerals protect these microorganisms from the radiation and desiccation. So you have all these different microbial ecosystems in these lakes. But why it's important to study these microbial ecosystems? Because the Central Andes present extreme conditions, such as, for example, low oxygen pressure, strong solar radiation. Because of this radiation, you have high evaporation rate, large seasonal and daily temperature ranges, and then hypersaline, alkaline, and arsenic-rich waters. And all these conditions resemble potentially the early Earth conditions. Therefore, the study of these microbial ecosystems can help us understand, for example, the limits of life upon extreme conditions, also can help us understand how early life on Earth evolved. And it could be used also, these microorganisms, for the search for life on other planets such as Mars. So now I will jump into the research. So over the last years, I have been studying different lakes and the microbial ecosystems in these lakes. But in particular, today I'm going to present you the study of Pozo Bravo Lake, that was possible thanks to the Cabana funding. So this lake is super important. Well, this lake is located, in Salar de Antofalla. Salar de Antofalla is the largest salt flat in the world, and it's located in the north of this salt flat. And the particular thing about this lake is that it harbors also the biggest and largest microbialite reef in the Central Andes. So you have, as you can see in the pictures, microbialites all over the shore. But in particular, the southern region of the lake presents a beautiful transition of non-lithified mats to lithified mats and then microbialites. So it's a super interesting area to study this formation of microbialites. So first we characterize the environmental conditions of the lake, and what we could see is that during winter, the temperature decreases and the water level increases, and therefore you have more dissolved oxygen and less ionic and mineral content. On the contrary, in summer, when the temperature increases and the water level decreases, you have less dissolved oxygen and more ionic and mineral content, especially of arsenic and manganese. This lake is super rich in manganese, and that is also a particularity. So talking about microbialites, the microbialites in this lake present different external morphologies. You can find, as you see there, tabular, domal, or even discoidal microbialites. But what is most interesting about these microbialites is the internal structure, which is called the mesostructure. It goes from a thrombolytic core to dendrolytic and stromatolitic layers. This is super specific, but don't worry. I just wanted to show you that it's also important to study the inside the rocks. So going to the mats that form these microbialites. As you can see in the image, normally these mats have an upper green layer followed by a purple green layer. So we studied these microbial mats through micro X-ray fluorescence in order to determine the distribution of elements in the mat. And basically the most important thing is that what we could see is that there is a manganese layer which corresponds to the purple layer, and then you see a sulfur layer which corresponds to the lowest layer. Then you can also see, for example, at the distribution of arsenic that is more concentrated at the bottom than on the upper part. And then what we did is with microsensors, we measured the oxygen and sulfide levels. And what we could see is that during the day and during the night. And what we could see is that during the day, there is a peak of oxygen that also corresponds to the green layer, basically because of the cyanobacteria in that layer, that disappears at night. And also we could see that the sulfide concentration increases at the bottom of the microbial mat. Then we perform scanning electron microscopy analysis of these mats. And what we could see is that there are a lot of diatoms, cyanobacteria, and other prokaryotic cells, which are forming aggregates embedded in a exopolymeric substance. And the important thing is that there are a lot of carbonate precipitation taking place above these aggregates. That suggests the participation of all these microorganisms in the formation of the rock. And we could also identify through these rocks different genera of diatoms, which we have also seen in other lakes, which shows that these diatoms are very well distributed among these different lakes. Then we studied the microbial composition of the water, the mat, and the microbialite in the different seasons. And what we could see is that the microbial composition of the water is completely different to the microbial composition of the mats and microbialites. But in the mats and microbialites, what we could see is that during the warm seasons, there is an increase in the phototrophs and a decrease in the chemorganotrophs. And on the contrary, in winter, the phototrophs reduce their abundance and increase the abundance of the chemorganotrophs. Well, this is just also we analyzed the alpha diversity of the samples, and we could see as that, of course, in the water, the diversity, there's less diversity than the mats and microbialites, and that the composition of these of the water is also different to the one from the mats and microbialites, which are more similar. Then what I wanted to do is to compare the genera from my microbialite with the genera of other microbialites all over the world. And what I could see is that it's super interesting because this shows you that most of the microbialites in the world present unique genera, and that is not shared between the different microbialites. Most of this is explained by the environmental conditions where the microbialites are present. But also this indicates that the formation of these microbialites is more due to the metabolisms present in these microbialites and not exactly the microbial composition of them. So that's why through the metagenomic analysis of Pozo Bravo microbialites, we obtained more than 150 metagenome assembled genomes. And of these genomes, we obtained the genome properties. Okay, basically this gives me an idea of what each genome the metabolic capacity of each of these genomes. And with all this information and all the other information that I presented before, I did this. This is a model of the microbial mat. So you have all the organisms involved in this mat, where are located in the different layers, the metabolic activities that they are carrying out, also the levels of oxygen, sulfide in this mat. And why I present you this? Because, and this is the last slide, because now this was like, so sorry, I'm like in charge of what it's called the microsig project in EMBL, which basically is to continue studying all these lakes and microbial ecosystems in these lakes, but in a really big scale. So we received funding from EMBL, and for example, before during my PhD, I could analyze more or less 10 different microbial ecosystems and 2 or 3 lakes. But now, thanks to this funding, we're gonna more or less analyze more than 20 different lakes, and in each lake we'll have more than like 3 or 4 microbial ecosystems in each one. So we have like more or less like 80 different microbial ecosystems that we're analyzing, and for each microbial ecosystem I would like to do something similar to what I did in Pozo Bravo, and to get that model of the mat and the microbialite in order to understand the metabolisms involved, the microorganisms that are there, in order to understand what's going on. Why? Because most of these microbial ecosystems present microorganisms that have unknown metabolisms, and that's super important for biotechnological approaches. So thank you very much for listening.
Thank you, Federico. One question. Kathy, you had one already. You can't have one every time. Okay, 2 questions.
Very nice work, very interesting. You got to a model. How can you test your model?
So now what I'm doing is isolating all those microorganisms, because we have a robot in EMBL to isolate them, which was not possible, of course, in South America. And the idea is then to evaluate how they grow in different conditions and see if, to prove, as I said, all those metabolisms, and try to find a new one in these isolates. So that will be the next step, to move to the isolates, like old microbiology.
Thank you.
Not someone else.
So very nice talk. So I couldn't see was the fungal component of the mats. Are there fungi there? Are there black yeasts, for example, that grow on the rocks and so on?
That's something that I always try to analyze, because there are no known like yeasts or like fungi that are like living in these extreme conditions. If I find one, I will publish in a good journal. But yes, no, I don't know.
So, but you do ITS sequencing and
no, I haven't. I just metagenomic analysis, but in the metagenomes, I have never found anything related to fungi.
Okay, thanks, Federico. Please make the most. I've got hundreds of questions for both of you, so I hope you got time during coffee to discuss. They're really great presentations, again, of global importance. So our next guest is Dr. Jose Pablo Jimenez from the University of Costa Rica. Has a doctorate from the Department of Forestry and Natural Resources in North Carolina. And we're going to hear about coffee genetic diversity. And if I can take a quote from SIP, imagine a world without coffee.
Well, first of all, thank you for inviting me. Thank you to Cabana NET. And before I start this presentation, I had 2 disclaimers, but after that talk, I have 3 disclaimers. One, this is not as sophisticated. Congratulations, that was a great work that you did. Second, this is just a small part of a larger project. So I'm just giving you a small picture of the first, like the preliminary analysis that we've done on this. And the second, and I'm a little bit ashamed of this, I'm not a coffee person, or I wasn't a coffee person before this project. I've grown a new appreciation for the beverage after working with this. I'm a plant breeder. I'm mainly on timber. So this will be my first forage into working with coffee. Yeah, let's see if we can go ahead and start now. There you go. All right, so coffee is a really diverse genre. If you're looking at the coffee genus, there's different types. It comes in different forms, colors, and flavors, if you want to think about it. However, there's just a small amount of the species there that have a significant caffeine content. That is what we're looking for, right? So despite this huge diversity in the species within the genus, there's only a few species that we actually use for commercial purposes. So this is a work from Hammond. What they did is they traced back the evolutionary history of caffeine content, right? And they determined, and I don't know if this has a pointer, maybe you can see it there. All right, so this is the clade where most of the species that have the higher caffeine content are. And there's believed to have evolved as a defense mechanism for predation, mostly on the equatorial climates, the humid climates, where you can find the different species, right? Now, the problem that we have with this is, again, that there's huge diversity within the genus, but we're only using a few of the species. This is from the World Coffee Research, which tries to map the different cultivars that we have, and want to call the attention to the fact that we're actually working with just 2 species. And one of those, Cafe Arabica, is actually a hybrid between Eugenioides and Canephora, Robusta coffee, right? So we're starting from a pretty narrow base because we're using a couple of species, despite the fact that we have several cultivars, and you can go to your supermarket, right? And you can go through the coffee aisle and see tons of different coffee brands, coffee types, but they're coming from the very same species, right? The very few species. On top of that, think that some of these accessions were introduced once or twice in the different countries. So we're starting with a few plants in every country, and we start to making crosses and so on. But then again, we have a narrow genetic base to start with, and that's applied to every plant breeder, right? Because we're suffering or we're risking genetic erosion, right? And just for the sake of people that might not know the concept of genetic erosion, it's just that loss of biodiversity that you can have when you have a narrow genetic diversity or genetic base. So this is from tomato, but the same applies for coffee. You can either have narrow genetic base, most of your individuals are near the mean, right? They look pretty much the same, or you can have a broad genetic base, right? You have some individuals that might produce more and some that produce less. The thing is that when you start selecting, and if you do an intense selection, let's say in this case, they put 10% selection, it's just a matter of a few generations that you narrow down significantly in your diversity. And once that you lose that diversity, you lose the resilience of your crop, you lose the capability to get more genetic gain, to be able to provide new traits or improve your productivity. And then again, that's a problem that all breeding programs have. So for all the plant breeders there, any plant breeding or tree improvement program at its core is a conservation program, because we need to conserve genetic diversity, because otherwise we don't have something to work with, right? We don't have material. So that said, we look into how we can assess or truly assess genetic diversity in coffee in this case, right? And there's plenty of data. Coffee is one of the major commodities. I think that after oil is the second most traded commodity. So there's plenty of information, plenty of data that has been accumulated on coffee diversity. However, traditionally it's been done with molecular markers, and each marker has its limitations, right? So if you're using marker data, you're looking at just a few parts of the genome, you're looking at allele frequencies, and most of those markers, or some of the markers that you use, while they're really good for population genetics and phylogenetics, they're neutral markers in nature, right? So it's hard to see the effect of selection when you're looking at markers that are not under selective pressure, right? So information is useful, but it's not as good as, and here I'm preaching to the choir probably, as genomics information, right? Looking at the whole genome will be the best that you can do. That's the gold standard, and people that work with genes can go ahead and look at the whole genomes. That's great. They're not that big of a genome, so you can sequence plenty of those and compare them. Coffee is not a huge genome. We're talking about 1.1 to 1.3 gigabases, but it still is large enough that it will be expensive to sequence whole complete sequence with a decent coverage of multiple samples, right? It's not as bad as pine trees, which 21 gigabases per genome, or metasequoia, which is, I think, the largest that we have so far is around the 40 gigabases. but it's still big enough that represents a problem. So reduced representation sequencing is the next best thing that you can do, right? And we're talking about RADseq or GBS, right? Genotyping by sequencing, allow us to reduce the complexity of that genome, but still looking at a lot of places within that and getting high throughput information at a cost-effective rate. And then again, this is probably basic to everyone that is here. You know how RADseq works, but just for the sake of people of the audience that might never done this technique, what we're doing is we're cutting the genome with restriction enzymes, and then we're just sequencing the small fragments within those that we cut and compare it along all the samples. And by doing that, we still get a fairly good representation throughout the whole genome without having to, sequence the whole thing. So I'll just go real quick here. The pipeline, we work with 160 samples from 16 different cultivars from Coffea arabica and 4 wild types. I'm using quotations there because they're really not that wild. We're including Canephora and Euphenoides, which are the parentals for arabica, and we're also including Liberica and another one that is part of the clade of the coffee plants that produce caffeine, but it's not the ones that are the most commercially planted. Canephora maybe being the exception. Robusta, it is planted throughout the world. In Costa Rica, we're mostly exclusively Arabica, but they're the different ones. And on top of that, I'm going to talk about 3 mutagenic treatments that were tested. Library preparation and sequencing was performed by Simba staff, so we're really thankful for that. And the pipeline, if you want to take a look, or we can discuss it later, it's pretty straightforward. What you will expect to go ahead and align your reads, generate a variant. The only thing that I'm going to call your attention is to the fact that we're using an old variant site and that we're using nucleotide diversity as our measurement of diversity, as a true measure of sequence difference. And we're using Pixie, which is a new algorithm that is really good at handling missing data, which is a blight that we have with RADseq data. We tend to have a lot of missing genotypes. All right, so again, I apologize. This couldn't find a better way or easier way to present this. It's really crowded there, but just bear with me on this and I'll walk you through it. You have the different cultivars here. This 4 over here is what we're going to call the wild types, so to speak. And what we're looking at here is a window of 50 megabases. So pretty much we're looking at each bar being a chromosome out of the 22 chromosomes that we have here. And we're looking at the nucleotide diversity, or the average nucleotide diversity of the pie, right, on for each chromosome for each of the cultivars. And what you're going to notice is that there's some chromosomes that tend to be a little bit more diverse than others, and that's directly correlated with the fact that some chromosomes are larger, so there's more possibility of changes, right? But what I really want you to look at is the difference between the cultivated coffee and the wild types. And you note a drastic, right, reduction in genetic diversity, nucleotide diversity. So there is a case for genetic erosion in coffee right now. This is just clustering all the cultivars and the wild type separated. And over here is really drastic, right? You notice the difference between genetic diversity at the chromosome level in each of those samples that you get there. So what can we do? How can we introduce new diversity? Well, the logical step will be to do introgressions, right? It's like to cross those plants with different genotypes that are wild types or that have a higher diversity, but also you can create that. You can induce diversity by inducing mutations. And that's what they did. This is a work with Dr. Andres Gatica, where he worked with the cultivar of Arabica, which is Catuay, is the most planted, one of the most planted in the country. And he used 3 treatments for mutagenesis. And we can talk about the details on that later. But what I wanted to bring you up is that all the treatments increase slightly the diversity, the genetic diversity that you have on your samples. And some of those, in particular, the treatment 2, produce a higher diversity, would you say, or introduce a lot of more mutations, especially in chromosome 6, 9, and 4 over here that you can see. So just to close real quick, because I know that I'm going over, future perspectives, we're working on a mutation finder and functional annotation on those mutations, on those mutagenic materials. That's a collaboration with Bradley Till at UC Davis. We developed a tool, a bioinformatics tool, that allows us to differentiate between natural variation and the induced mutations and functionally annotate that mutation so we can know what potential effect might have on the phenotype. We're working on a GWAS, so we can screen for these mutagenic cultivars for coffee resistance, for coffee rust resistance. This has been done at the lab level on control environments, but we're taking it to the field. Those plants are already in field trials there. And of course, we have to think about integration at some point. We'll have to introduce new variation by control crosses with different coffee cultivars, hopefully not losing the characteristics that we already have achieved. Acknowledgement, the University of Costa Rica, just a small disclaimer as well. The project is under University of Costa Rica. Dr. Andres Gatica is the PI, but my home institution is Tecnológico de Costa Rica, is another university that I work at. We also have funding from Cabana. We really appreciate the work from CIMA staff, and as well as data that we can get from the NCBI and the World Coffee Research Center. Thank you.
Great, thanks very much. So another example. Of some regional collaboration, which is great. And again, with climate change and all the rest of it, I'm sure this research is important. One question.
Thanks, Jose. That's a great job, a great research. So for GWAS, for rust resistance, that's pretty hard to do for many reasons. Why don't you try some markers that for caffeine that you could start with this. At least you have some of the ones that have a high caffeine content against the Arabica ones. So perhaps for GWAS that would be a better start than with the rust resistance, which is pretty hard.
Yeah, I completely agree with you. It is a problem. However, it was one of the main objectives of the project to look at rust resistance. So that's why we have it there, even though I know it's not an easy job to do. And regarding caffeine, well, that's pretty much well typified. So we could look at that, and that's part of what we're doing with the annotation of the mutations, right? Looking at see if any of those mutations are actually in the genes that are related to coffee content and how they could potentially affect caffeine production and coffee quality in general. But yeah, it's something that definitely worth looking at.
Okay, thanks very much. Okay, our next speaker is Dr. Carlos Modinuti, who we know very well from the Cabana project. He's contributed huge amounts to our project. He earned his PhD at in pharmacy and biochemistry at the Department of Buenos Aires, works with Adrian Trzymski. He's now working in drug discovery, but during the Cabana project, he worked on yerba mate, and I think this is a labor of love.
Okay. Hello, everyone. Well, I'm going to present the yerba mate project. This is an honor for me because I was born in the region in which the yerba mate farming grows, and also study in the university in which is central, and basically grow up in a region in which yerba mate is everywhere. So for us, for people for the region, not just for Argentinian, this project was very important. Was encouraged. It was a great initiative from the government at the beginning, trying to create this project. And let's next one. Okay. Well, yerba mate is a tree, so it's not a plant. It's a tree that grows in the region in the south of Brazil, in the north of Argentina, just in two provinces, because there are specific conditions for the yerba mate growing. It grows in a field rich in iron that just is found in this region. So you cannot cultivate yerba mate outside this region. And I was born in the red dot and studied in the green one. So I moved to the yerba mate region. And Argentina is the highest producer of yerba mate, and sharing with Brazil and also Paraguay. But not only in Argentina we consume yerba mate, also is very popular in Syria. 70% of the products that we export goes to Syrian market, and now becomes very popular in Europe. So I think it was an effect of the World Cup, in which was a lot of publicity. So it's consuming in South American countries. Well, the reason is because it's cultural, but also because it has very good properties, like antioxidants properties, antimicrobials, anti-inflammatories. It's diuretic, hepatoprotective. It has minerals important, but I think it's very popular also because it's a stimulant. We have a lot of caffeine. And for that reason, football players consumes a lot. But this is not just a story from, yerba mate is very good and so on. There are a lot of publications in which there was proven the effects of yerba mate, for example, in modulation of gut microbiota, in the prevention of obesity, and also in the prevention of some disease like Alzheimer or neurodegenerative disease. And also, it's not just the consuming of yerba mate with the traditional way, using the specific device. Also, there are a lot of product derivatives of yerba mate, like drinks, soaps. You have a lot of products derived, and it was a surprise for me that in Europe the drinks based on yerba mate are very popular, specifically in Germany. This is one of the most popular drinks in Germany, and it's based on yerba mate. I don't know how they import the yerba mate, but they produce a lot. So there was previous works in yerba mate. There was a lot of publications, but the most relevant and important was the transcriptomic of the yerba mate, in which it was the first approach to try to identify the genes that are involved in these amazing properties. But transcriptomes has limitations. This was published in 2014 for people from the university in which I studied. This guy was my professor, actually. And well, the final conclusion was the first draft of the genes, and also the model of the chloroplast. But it was not enough, because, as I previously mentioned it, yerba mate is a tree. So if it's a tree that takes like 10 years to produce. So it's and before the 70s, it was just a process. They were not extensive farming. There was They started in the 70s, and every process of genetic improvement took around 10 years. And that is a limitation. And also basic things like the sex identification in plants took around 10 years. So it's very complicated for breeding programs. So in our approach, we obtained the samples from yerba mate, and we apply two different approach, Illumina sequencing and PacBio sequencing. And we were able to assemble a very a nice version of the genome. And one of the, I want to mention just some findings of the research. It was the caffeine biosynthesis pathway identification. We were able to identify 3 different enzymes that are involved in the caffeine pathway, and also the crystallographic structure of the caffeine synthase. And this is maybe one of the most important, because we annotated and deposited in public available repositories, enable to the researchers to access to this information and work with. It was very important for the researchers, mainly in Argentina, Brazil, and Paraguay. And if you want to take a look to the more findings from the genome yerba mate, you can access to the preprint version of the paper. And for me, one of the major impacts of this project, in which involves Cabana particularly, was the capacity building development in the north of Argentina, because these people that is very good working with the plant and doing very nice research, they were not able before the involvement in Cabana segments to do it by themselves. They also always collaborating. And now we trained Maximiliano, that is researching in that region, and he started their own group as bioinformatics in there. Finally, I want to thank the collaborator that participated. It was for the publication, it was University of Buenos Aires and the University of Northwest in Argentina, and also was people from Vale Institute in Brazil and Illinois University in the US. And also for the funding provided for the National Research Council and the government of Argentina. Thank you.
Great, thanks, Carlos. Nice to see the project, all the work that you've done, brilliant. Any questions?
Are there any important differences between the caffeine synthesis pathway in coffee and the caffeine synthesis pathway in yerba mate?
Yes.
Yeah.
They are the pathway that the yerba mate use is different from coffee, and also the tea, Camellia sinensis. It's an alternative way, but it was previously reported. It's convergent evolution, the conclusion, yes.
One more question for Carlos before coffee.
Very nice work. So I was wondering, so seeing the coffee talk before, are there many cultivars from yerba mate? Are there wild relatives? How does the genetic diversity and the diversity overall look?
Yes,
like
they are kind of
mate?
wild cultivars, and they are. We have a national institute that for farming that create these genetic specific variants that allow the, for example, macro propagation, because you know if you must spend too much time to grow the seeds and propagate it, and there are some commercial and private varieties and some publics that are provided for the national institutes.
Okay, thanks very much. Can we have coffee, reconvene at 10:25, and then we'll just be 5 minutes behind schedule. So our next speaker is Valeria. She is a researcher at INTA. She has a BSc in agronomy, an MSc in soil science, and a PhD in biological science. We've had the pleasure of seeing her in Cabana. She's been working with Cabana NET, and Valeria is going to talk to us about exploring soil microbiomes of contrasting agro-ecosystems in Latin America. Thank you.
Hello, good morning. Thank you for having me. Okay, I will try with this. Okay, well, I will talk about the projects that we conducted in Cabana. We can say Cabana 1, Cabana NET, Cabana 2. We were investigating soil microbiomes with one main idea, which is food security. Food security and the challenges vision to produce with lower amounts of fertilizers, with new pests and pathogens, with climate change, and reduces lands to expand the agriculture. This is the mission of our studies, to identify the microbiomes. This is the soil bacteria and fungal communities, which can help to promote the agriculture. And this is not just one idea that we just came up. This is a claim, a global claim to produce more by 2050. It is estimated that we will need to duplicate the agricultural production, not only the quantity, but the quality of the production. So we need to identify how to improve the use of the resources, just the fertilizers and water production, and maintain the quality, even though all the restrictions that we can have due to climate change. Well, in the first project that was funded by Cabana, we explored microbiomes in 3 contrasting environments, in coffee, Colombia with Marco, in Argentina in long-term experiments, which are typical of Argentina, in Peru, it was an amazing survey of maize cultivated in an altitudinal gradient in Andes, pre-Incaic terraces, which are, well, amazing results are coming from there. And the idea of this project was not only to generate data, but also to build the capacities of people in Latin America to analyze this data and to comprehend the information and the importance of the information that we are producing with this type of analysis. So the main idea was to focus on food security, but also to promote the capacity of students to analyze the data. Another issue was that there is certain constraints about the differences in results according to the labs that produce the results and the people that analyze the data. So we create a protocol to analyze just the exact way from taking samples to produce the data just in the same conditions across Colombia, Argentina, and Peru. We adjust a protocol to extract the DNA and then sequencing in Argentina. It was a great effort to do the sequencing in Argentina for many reasons. And the bioinformatics was done in Colombia, Argentina, and Peru with secondees. We had 3 secondees, Lorena from Mexico, Over from Peru, and Victor from Colombia. well, and Nati Pimbiso from INTA was collaborating also in this analysis. Okay, this is, so for us, I always show this picture because after the pandemic, there was a great success to having all the resources to do the sequencing, all the sequencing from Peru, from Colombia and Argentina, and I conducted myself. So I learned how to do a library and how to sequence, so I'm proud of that. And I thank Cabana for it. Well, the people there, Mariam, Pablo, they were really helpful. They were struggling with the pandemic and the COVID sequencing, and they, well, gave me time and space to do the analysis for Cabana. And it was, well, the Se Largó Cabana, well, Cabana Argentina start. Well, and the results, I just will summarize the results and the main ideas. Food security was thought or was planned as crop practices that were sustainable. And we focus on management agronomically the crops to produce more, to produce in an economically profitable way, but also taking into consideration the soil health, which is having the appropriate soil communities in microbiome. Okay, so the main idea was to compare three situations. This lines is one situation, which is cultivate year after year the same crop, which is soybean. During winter, you have nothing cultivated, and then soybean. The next year, nothing, and then soybean, and over again for many years. And then there's another option, which includes other species, that's maize, then soybean, and then the double crop with wheat and soybean. And this option, which includes always living roots in the system, year after year. So we had the idea, and we proposed based on evidence that we can have, if we can measure the health of the system, this could be the more healthy system because they have roots and they have the soil cover, so we don't see soil in the river, as Ian told us. So in this system, it could be the poor quality because we are cultivating the same crops, we leave the soil without any roots. So it could be not the best situation. So what we found was amazing. And I just show the conclusions, which is in this situation, we found a community with predominance of pathogens. And in this situation, we found a community, microbial community, dominated by symbiotes. So microorganisms, we can improve the tolerance of plants to water stress, to heat stress, to conditions of low fertilizer inputs, etc., etc. It was an amazing result, and we are very proud of it. And it was found in bacteria and in fungal communities. So the same, but adapted for another situation is the Colombian coffee production. The same idea of maintain crop diversity and crop production based on sustainable manners. So Marco showed yesterday that they sampled these three locations, which are with never been sampled before for studies in microbiomes. So we studied fungal and bacteria, and here you can see just the production. We have this that picture without just only coffee. It's like a monoculture, only coffee species. And then here is an amazing picture that I found in the page, that website that created our secondee. This is the agroforestry, which is cultivate the coffee with plants, with native plants. And these plants allows the conservation of biodiversity, birds, mammals, and all of the animals and organisms which are not the target of our investigation, but can also improve the sustainability of the system. And you can see here the second side here is completely different from the others, and this is agroforestry, and the others were newly established coffee farms without forest almost. And well, why was that such a differences? And well, we found this in agroforestry, a higher abundance of very helpful insect pathogens which can control pests without using insecticides. And we found this astonishing abundance of this yeast, and we then would like to continue further investigations because this yeast was found in soil samples, but there is new evidence that it can be found in grains and it can improve the organoleptic quality of coffee. So maybe agroforestry is not only allowing the conservation of biodiversity, but also promoting best quality of coffee. Okay, where we wonder what next. Well, now we have the second project which I'm leading. In Argentina, we took samples. This is the general situation. In 2023, we could do the sampling, DNA extraction and sequencing. And next year, we are expected two secondments, a workshop organized in Argentina and publications. In Costa Rica, with Andres Gatica, we will compare again the coffee production with agroforestry and less sustainable practices. They already done the sample DNA extraction and sequencing, and now we are having a workshop the upcoming week and publications. In Peru, with Jan, we will study the same agronomical practices, aiming to promote soil health to produce potato in an amazing experiment with all the experimental design and working with farmers to promote the use of sustainable practices based on the results of bacterial and fungal communities. Well, we have selected the second D, which hopefully is starting next year, and we're expecting publications. The main outcomes of these projects for this, at least for this year, is the workshop. We had 85 postulations, selected 10 from Argentina, Peru, Colombia, Ecuador, and Mexico. We are very proud of it. And the microbiome project also selected one secondee to do a secondment in Peru and Argentina for 6 months. Thank you very much.
Questions, very quickly.
Very quickly. So are you sure the fungus you found in the coffee plantations is Metarrhizium anisopia, or is it Metarrhizium robertsi? Because robertsi has some implications in direct benefit to the plant in terms of nitrogen metabolism. So there are two species of Metarrhizium that interact with plants. One is anisopia, that is basically a pathogen. But there is robertsi, which is very closely related, and it interacts directly with the plant and improves nitrogen metabolism.
Okay, well, the databases for fungal taxonomic identification of metabarcoding has several, well, a little bit constraints related with the resolution of the results. We could identify Metarrhizium anisopli, which produce the entopathogenic control of pests, and a large set of species which were identified at the genus level, not at the level of a species.
And the last one is just how can you handle then the microbiome in coffee plantation? Because in cultivated crops, it's easy. You change the practices, but with the coffee, what would you do?
Oh, you can change the practices. You can improve the, allow the coexistence of other plants, not only coffee, increase the plant diversity in your plantation. If you can, you should incorporate agroforestry, definitely, because this was the main change in the soil microbiome, the agroforestry, allowing the biodiversity there.
Just to clarify about Metarrhizium, it's not surprising that we find it because it's been widely used in Colombia to control coffee berry borer. So, okay.
Yeah, no, I'm going to have to stop there. Sorry. Great example again of sort of cross-border collaboration with institutions in Latin America helping each other out. So thanks very much. Okay, our next speaker is Macarena Soledad Concha Tolosa, who is this a sorry? Virtual. Okay, great. It's a virtual presentation. Macarena was a secondee on the Cabana project in Costa Rica. She has a background in hematology and a master's degree in science. She worked as a teacher at the Santo Thomas University in the School of Medical Technology from 2021 to 2022. Currently, she is carrying out research collaborations in the bacterial enteropathogens laboratory directed by Dr. Luis Costello, and this is a virtual presentation. Macarena, are you there?
Buenos días. Ya, buenos días a los presentes. Mi nombre es Macarena Concha Tolosa y a continuación les voy a hablar sobre los patrones respecto de la circulación del virus SARS-CoV-2 en América Latina. Este es un proyecto el cual ha comenzado el año 2023 y que tiene como fecha de finalización el año 2024. Ahí está. Este proyecto Cabana se titula como patrones genómicos, socioambientales y de capacidades respecto de la circulación del virus SARS-CoV-2 en América Latina hasta el año 2024 para apoyar la vigilancia epidemiológica utilizando un enfoque multivariante. El periodo de duración sería entre septiembre del 2023 y febrero del 2024. Como coordinador encontramos al doctor José Molina Mora. Hasta ahora se ha avanzado con este trabajo, pero todavía queda bastante trabajo pendiente que se espera trabajar fuertemente el próximo año cuando se realice la pasantía de manera presencial en la Universidad de Costa Rica, donde también vamos a trabajar con nuestros colaboradores que serían el doctor Darío Fernández Soporto, Alfredo Heriberto Herrera Estrella y Luis Collado González. Mi papel dentro de este proyecto es como secondie. Como se dijo previamente, soy investigador que colabora con la Facultad de Microbiología de la Universidad Austral de Chile. Tengo formación de tecnólogo médico con magíster en microbiología y yo postulé para ser secondie de Cabana en junio del 2023. Dentro de las actividades está un entrenamiento en bioinformática y lo que se ha realizado hasta ahora, que es el análisis de indicadores socioambientales y de casos de infección del virus SARS-CoV-2, pero todavía queda trabajo por realizar. Para comprender un poco el contexto de este proyecto, la enfermedad de COVID-19 ha afectado no solo a los servicios de salud pública, sino también otras actividades como actividades sociales, económicas, ecológicas alrededor del mundo. Este virus ha infectado a más de 753 millones de personas, con al menos 6.8 millones de muertes hasta el año 2023. Respecto a la pandemia en Latinoamérica, esta ha sido una de las regiones más afectadas. Hay que tener en cuenta que hay un lento crecimiento económico, que hay el proceso se ve limitado de los indicadores sociales y que también hay una amplia diversidad ecológica en esta zona. Por ello, cuando hablamos de programas locales de vigilancia de las enfermedades, estos dependen mucho de las decisiones y capacidades de cada país. Aquí juega un papel importante la iniciativa Cabana, ya que esta iniciativa nos ha permitido tener un mayor conocimiento bioinformático en esta región, como también del virus SARS-CoV-2. Previamente, la iniciativa Cabana permitió realizar un estudio, el cual contribuyó a la vigilancia genómica del virus SARS-CoV-2 en América Latina. Esto permitió comprender cuál fue la dinámica de la pandemia en esta región a partir del análisis de los genotipos en ciertos países. Entonces, este se centraba más que nada en la diversidad genómica del virus SARS-CoV-2 en estos países de la región. Pero hay que tener en cuenta que también el virus SARS-CoV-2 genera una infección, una enfermedad infectiva, donde tanto la diseminación y propagación como las manifestaciones dependen de la actividad epidemiológica, es decir, del agente, que es el virus SARS-CoV-2, del huésped humano, con sus comorbilidades y factores genéticos, y del medio ambiente, ya sean condiciones ambientales, físicas, sociales, entre otros. A partir de esto surge este nuevo proyecto, el cual tiene como finalidad no solo actualizar sobre la vigilancia genómica del virus SARS-CoV-2 en América Latina, sino también establecer ciertas relaciones entre las características del huésped con indicadores socioambientales y la capacidad que existe en la vigilancia genómica del virus SARS-CoV-2. El objetivo general fue determinar los patrones genómicos, socioambientales y de capacidades asociados con la circulación del virus SARS-CoV-2 en América Latina hasta el año 2024 para apoyar la vigilancia genómica a nivel regional utilizando un enfoque multivariado. Los objetivos específicos fueron describir los patrones epidemiológicos entre los indicadores socioambientales y los casos de infección de SARS-CoV-2 en América Latina mediante un análisis de la base de datos de indicadores demográficos, sociales y económicos con un enfoque multivariado. También determinar los genotipos y los procesos evolutivos del virus SARS-CoV-2 circulantes en América Latina mediante el análisis de secuencias genómicas y de condiciones del huésped a partir de bases de datos para apoyar la vigilancia genómica regional. Y también evaluar la vigilancia genómica mediante las capacidades de secuenciación en América Latina, usando una estrategia multivariada con datos asociados a la intensidad de la secuencia, al envío desde la recolección y el tiempo de detección de la variante del virus SARS-CoV-2. Entonces, la estrategia general está basada en la utilización de bases de datos públicas. Para los patrones socioambientales y de capacidades, lo que se hizo fue buscar los indicadores demográficos, sociales, económicos y del virus SARS-CoV-2 de 24 países en bases de datos. Estos indicadores luego se les realizó una correlación donde permanecieron y se siguieron utilizando aquellos que tenían correlaciones moderadas a fuertes, para posteriormente ver una asociación entre ellos. Respecto a los patrones genómicos o genotipos del virus SARS-CoV-2, utilizamos dos bases de datos, o se planea utilizar dos bases de datos. GISAID, que nos permitirá obtener las secuencias y los metadatos del genoma SARS-CoV-2 de América Latina, y Pangolin, donde obtendremos la frecuencia de los linajes entre los países. Estos datos generarán un árbol filogenético completo. Entonces, lo que hemos avanzado hasta ahora ha sido en los patrones socioambientales. Como dije previamente, se buscaron estos indicadores para 24 países, de los cuales 11 corresponden a países de América del Sur, 8 a países de América Central y 5 a países ubicados en el Caribe. Estos indicadores fueron buscados en diferentes bases de datos. En la presentación se muestran algunos ejemplos, donde finalmente se seleccionaron 334 indicadores socioambientales y 4 indicadores COVID. Luego se hizo una correlación de Pearson para ver quiénes tenían correlación positiva o negativa, donde finalmente permanecieron o se siguieron utilizando solo 89 indicadores socioambientales, los cuales interesaban saber cuál era la asociación con los indicadores COVID. Por lo tanto, se hizo un modelo lineal generalizado para ver dicha asociación. A continuación se pueden ver los datos preliminares que hemos obtenido hasta el momento. Se observan los indicadores COVID por un lado, y por otro lado se muestran aquellos indicadores con los cuales tenían una relación, como también el tipo de correlación que presentaba, ya sea positiva o negativa, siendo estos últimos visualizados de color rojo. Entonces, por ejemplo, podemos ver que por los casos COVID por población se asociaban, tenían una correlación negativa con la temperatura. En el caso de las muertes COVID, era una correlación positiva con el índice de desarrollo humano, o las muertes COVID, según casos COVID, tenían una correlación negativa con la edad media de escolaridad. Entonces, como dije previamente, estos son datos preliminares, todavía falta realizar un análisis completo y también seguir trabajando en este proyecto, lo cual se planea realizar el próximo año de manera presencial en la Universidad de Costa Rica. Mis agradecimientos a las entidades que han permitido y que permiten realizar este proyecto, como también al profesor José Molina Mora por su guía durante la realización, la profesora Rebeca Campos y Luis Collado, los cuales han colaborado en el proyecto. Acá se observan los contactos. Muchas gracias por su atención.
Gracias, Macarena. Any preguntas? Any questions? it's not a question, but I'd like to add to this because this has gone quite a long way. So nobody knew COVID was going to come. This is giving us an opportunity to prepare for the next pandemic, which will come. Some other work that has happened because of Jose and Alejandro is that we've actually got a network of employees from reference labs in Latin America that we've put together, and we're now identifying their training needs and finding different trainers, not just EBI, but different trainers that have budgets to make sure that the people working in the reference labs in Latin America are equipped to be able to make the right decisions when the decisions need to be made. So this was a great start, and the science is continuing. And 2 years ago, we didn't know that we'd be doing this.
I have a provocative question. If all of the data for SARS-CoV-2 was available in the open access databases, the raw sequence data, the access to the clinical variants, the kinds of things that Gabby was talking about in Recoded yesterday, what else would we be able to do on top of what Macarena has achieved? And maybe that's a rhetorical question, something to think about.
Do you want to try and answer that, or do we leave it rhetorical? Okay, we'll leave it rhetorical. Okay, round of applause again, please. Well done. This has been a great piece of work. There's a lot. Okay, so our next presenter is Alejandro Reyes. Now, Alejandro was on the Cabana project. We have a wonderful relationship with UniAndes. Because of Alejandro, we have a training program that we do every year with UniAndes, and this has now grown so that the University of Costa Rica is involved in the training program. This is giving opportunities for lots of people in Latin America to see the kinds of services that we have at EBI to be used. And I must mention some of his bio, which is quite amazing. So Alejandro is an associate professor at the University of Los Andes, leading the research group in microbial ecology and computational biology, also the chair of the undergraduate program in biology and microbiology. He is the group leader at the Max Planck Tandem Group in computational biology and holds the position of adjunct assistant professor at Washington University School of Medicine. And Alejandro is going to talk about exploring the human gut microbiome, diversity in Latin America.
Thank you, Ian. Okay, so this was not a Cabana project. This is a project that started with Cabana NET. So this is still mostly of it is what we want to do more than results that we have obtained this far. So this is exploring the diversity of the human gut microbiome in Latin America, focus on the population at nutritional risk. So in general, the funding part of this project started because, as we heard yesterday from Hilary Brown, a lot of studies have been done in human gut microbiome, and what they have seen when they begin comparing most of the studies is that we have already almost saturated the bacterial diversity of the northern part of the globe. However, when we look at the southern part, like African, Latin American population, they're really underrepresented. So different initiatives have arisen in order to better characterize what is happening in the south part of the world in terms of gut microbiome diversity and what can that tell us. And there has been this very interesting effort called Latin Biota that Hilary mentioned yesterday that has been surveying the healthy population across Latin America. And then within the Cabana NET network, we begin thinking is like, we want to participate in this, we want to bring some samples and begin also extending that into other parts of the country. And we, after some thinking and pondering, we decided that we want to focus in our countries there's large populations that have non-Western diets. We know that obesity is a huge epidemic on the Western diets, and we have understood a little bit on how that's caused because of Western diet. But in our populations, we have at-risk population of obesity that do not have necessarily exposed to Western diet. So it will be interesting to see what particular aspects of their microbiomes are there that they are putting them at risk, or how their microbiomes are changing that are putting them at risk of obesity while not having a Western type diet. So that's something that we say, okay, that's something that we can study. And now we want to sample healthy adults at risk of malnutrition to characterize their microbiomes. This was also a little bit of a strategic decision by the fact that Cabana NET is just a 2-year project. It had to be short. If we wanted to deal with patients or populations with particular characteristics, that may mean more permissions, more ethics committee, and etc. when we begin getting into other populations. So that was going to be harder to get within the time frame that we had. The idea is, in several aspects, assemble new bacterial genomes. So this is not going to be metataxonomy. This is going to be full shotgun sequencing. So we can get new bacterial genomes, characterize a little bit better that diversity that we have in Latin American countries, identifying new genes, new functions, and in particular, associated with antibiotic resistance that we know is highly prevalent in our populations. So participating countries, we have Colombia, Costa Rica, and Argentina. In Colombia, we have Uniandes with me representing. These are just the PIs involved. There is more people involved in the project, but this project gave us the opportunity to bring new members into the collaboration. So we brought in CIDEIM, that is another Colombian institution with Maria Laida Gomez. And in Costa Rica is UCR, led by Rebecca Campos. Argentina is present with UBA with Adrián, and we also bring another institution that is Universidad Nacional de Misiones with Karina Salvatierra. So I'm not going to read this in detail due to time, but this is a little bit of the inclusion-exclusion criteria. So inclusion criteria, we're focusing in adults, in particular in the Colombian cohorts. We're looking to Afro-descendant population in risk of malnutrition as defined as body mass index of more than 25. Remember that malnutrition, obesity is also considered malnutrition. It's anything that deviates from healthy nutrition. So it can be overnutrition or undernutrition. So obesity is also defined as malnutrition. Any gender has to be in the region, voluntarily participating in studies, signed informed consent, and some exclusion criteria, different diseases, using medication, and so on. In terms of the procedures, we are doing some medical evaluation, evaluating the inclusion and exclusion criteria. In the case of Colombian women of reproductive age, we're also going to do a pregnancy test, since that's one of the exclusion criteria. They're going to be able to fill a survey that has socio-demographic data, nutritional data, and then we'll do the specimen collection, stool samples for intestinal metagenome, and some regular clinical lab. In terms of the Colombian kind of workflow, and more or less right now, here's where we state, we have the CIDEIM lab for DNA extraction with Maria Laida and Mariana. Then we have the CIDEIM clinician staff with different clinicians on site and the town that we're going to sample. We have the manager of clinical data, but a person that is expert. So this is more or less what has been in place now in order to collect the samples. Then all the samples are going to be sent to the Sanger Institute, to Trevor Lawless lab, in collaboration with Lighting Biota for sample processing. And then data processing and analysis is going to be done mainly at Uniandes with the help of 2 secondees that are going to be involved in this process. And then these secondees are also going to be doing some training on the data processing. What we have done so far, so in general, and in the other hands, we'll go into more detail of this part. We are in most of the cases around this part of the study because of signatures of the specific agreements. We have to buy all the collection sample kits. We have to buy all the extraction kits, and that has been delayed as well. We have to present the projects to all the ethics committees from each of the different countries. That create also some delay. So because of all those delays, we are currently still in the getting everything set up, and we have already agreed. So for example, it's very important data, this social demographic and nutritional data, because that's the metadata that we're going to be able to use to compare and contrast and gain more information from the sequencing data. Also, these questionnaires were carefully designed in order that they will be in agreement with all the surveys that have been done with Latin biota, such that when we generate all the data that we're going to generate from this pilot study, that is 50 patients, 50 individuals from each country, then we'll be able to compare with the approximately 600 samples that Latin biota has already generated. So in order to be able to compare those data sets, we have to make sure that all the metadata is consistent and in agreement between the different studies. So that's something that we have done already. The participant recruitment, so we have done in Colombia 2 different strategies: active search, supermarket, pharmacy, market squares, vegetable fruit stalls with different flyers in order to get people interested in volunteering participating, and some passive search when they go to the clinic or to the office and because of the advertisement. We have generated already some instructional booklets, in particular on how the sample collection must be done in Spanish so that the people will understand how to properly collect the samples. This was something very important because we are recruiting patients not in major cities, so it's a distance away from laboratories. So collecting the sample in the right way and being able to preserve it in the right way is very important. So these collection devices have preservants, DNA preservants. So we know that as soon as the sample is collected, it will go into the preservant and it'll be fine and stable even at room temperature for the time needed to get it into the lab and then to be frozen down and processed later on. We have the surveys. They are going to be filled when the patient goes into the lab by a clinician. Is going to have nutritional information, dietary frequency information. So we'll know what type of foods they eat, how frequent they eat, and those will be connected with the microbiome diversity as well. And in terms of the strategy of recruitment, we have active search and passive search, as I mentioned. Then we evaluate the inclusion-exclusion criteria. They fill up the surveys. They are then appointed for collection, a medical evaluation, collection of the samples, and then, we provide them with the sample collectors, and then they will collect it in their own houses. And then it will be, somebody will go to their house and collect the device and bring it back to the lab. So that's what is set up right now. We are, in case of Colombia, we're still waiting for these kits to arrive because they have some delay in arriving. Everything else is set up. Participants have been invited. All kind of this part is already in place. We're just waiting for the collection sample kits to arrive so we can start collecting the samples. Our idea is that January, the latest, we'll be shipping those samples to the UK for DNA sequencing. Hopefully, somewhere around February, March, we will start analyzing the data. So the next steps precisely is once samples are collected, send those to the Sanger Institute for sequencing and then the data processing at UniAndes. We're planning to use a combination of the Magnify EBI platform, the computational pipelines developed for Latin biota. As I mentioned, it'll be very important for us to perform comparative analysis using the Latin biota data. And with this, we're planning this as an initial data set, just 50 samples from each country, to generate enough data for future grant applications in order to go to different populations with larger grant with a little more time. We should be able to now go to actual patients or kids or other populations, and then train within the team and external on the data analysis workflows. And thank you very much. And these are all the institutions participating.
Thanks, Alejandro, and thanks for everything that you've done for Cabana and Cabana NET over the last, what is it now, 6 years nearly? It's great work, so well done. Kath, you have a question.
I'm not normally this vocal at meetings. I just get very excited at Cabana NET meetings. So this is a very naive question, Alejandro. You did mention AMR at the beginning of your talk. So will you be able to extract data about the mobile elements in the
Yes,
microbiome?
but we know there are pipelines. We have also developed in some previous work I've done pipelines in order to detect antibiotic resistance genes on shotgun data. We assembling the whole mobilome, like plasmid, large plasmids, and so on, it's a little harder with short reads, but we can get to assemble full resistance genes and resistant mechanisms. So that will be able to survey. And we know these at-risk populations, they have the tendency to auto-medicate with antibiotics.
Right.
So based on some previous information we have, we know antibiotic resistance in these populations is pretty high. So we want to a little bit address how high and what type of antibiotic resistance is present.
Okay, and are you capturing information on antibiotic use in that?
In
Brilliant.
the surveys, we have
Brilliant.
all that
Okay.
information.
Okay,
In
fantastic.
terms of disease, antibiotics, probiotics use, in terms of other aspects of their medical conditions that could lead us to know a little bit how possible is that they may have some antibiotic.
This is a really, really cheeky follow-on question, and I should actually check with Thea before I ask this question. Would including a Caribbean country in this study be of interest? And would it to Thea? Is she in the room? Yes.
I think definitely. I know Latin biota, so let me just go briefly back. So Latin biota, all what is in red are members, so I don't see any red in the Caribbean. So I am sure that both for Latin biota led by Gregorio, but the Trevor Lawley Laboratory, and for us, it would be very nice to have.
We should talk about that.
Extend and this is definitely what we're trying to do by the seeding on getting new information to show that Latin American bacterial diversity is larger than what has been measured in order to justify it to keep on sampling on other countries.
Okay, super. Thank you.
One more pregunta.
Hola. Congratulations for the presentations. I'm sad because my country is not there in the map. Sorry. I'm Bolivia, right? And Venezuela. Okay, but I am talking about Peru. But are you thinking any collaboration with any institution in my country, Peru?
So in terms of Peru, we have good collaborations. Actually, so this is Latin Biota 1. Latin Biota, it's already in a second round. And Pablo Tsukayama from Universidad Cayetano de Heredia, he is part of Latin Biota 2. So I know that some of the samples that he has been collected, they are not going through the Cabana NET project, but they are going directly through Latin Biota to get sequence data.
Okay.
So there's a second. So the first round of Latin Biota was just healthy individuals from a subset of countries that participated. Now they're beginning to focus on some patients and disease population and adding more countries into the project. And that's why we also want to connect Cabana NET with Latin Biota to add more samples into that.
Very nice presentation, Alejandra. Thank you. This sounds like a very exciting project, so I'm excited to hear how it all goes. Question about building capacity for like the sample processing and retaining data within Latin America. What are you thinking about that? Obviously, it's a huge lift in terms of server capacity, doing the sequencing itself, but what does that look like in the future, either of this project or moving forward into other projects?
We're a little bit discussing that earlier this morning with Kat at breakfast, and that's something I keep thinking. I definitely think that bringing all the data analysis capacities and training on how to properly analyze, but I do think that there's been huge advances like Magnify, and we have somehow collaborated with Rob Finn, and we know all what has gone into building Magnify. I think given that those pipelines exist and are there, it's not worth trying to reinvent the wheel in our countries. It's better just to use that infrastructure. I'm on all in as soon as we get the data deposited, ENA, but then do have the capacity of processing and analyzing those data, that if we want, we can download data, look for more things, look for more genes with that. And we were discussing out of fishing out certain specific genes out of the data set. We should be able to do that. We should have the capacity in our countries to doing that sort of analysis without necessarily needing the physical infrastructure to be in our countries. But that's a very open discussion I would like to have.
Great, thanks, Alejandro. So we should celebrate that this is actually, so Latin America is part of an international network doing this project. And when we started the Cabana project, not that we're responsible for this, we were concerned that actually Latin America does tend to be left out of these international consortia. So hopefully there'll be more of that going on. So thanks a lot. Okay, so our next speaker is Adrian Tojansky. He will be talking about target pathogens from genomes to drug discovery in neglected diseases. Adrian was part of the Cabana project. We've had the pleasure of visiting Adrian in Argentina and trying to understand a little bit more what's going on. Part of his bio, if I may. Yeah, so where do I start? I'm trying to winners of the world. Adrian would like to let us know that Argentina won the World Cup. And in his spare time, he is professor in charge of the structural bioinformatics lab in the School of Science at the University of Buenos Aires. He was the director of the Argentinian bioinformatics platform and director of the Center for Interdisciplinary Science. Over to you, Adrian. Thank you.
Thank you. So today I will talk about a project that started in my lab long ago, and now we are doing the next part based on one of the projects of the Cabana NET. So our main idea here is go from the genomes to drugs, and the idea is to be able to identify new targets, especially in neglected diseases. So what we do is to develop bioinformatics tools in order to identify these new proteins and then be able to identify new lead compounds that allows us to develop. In particular, next slide. So in particular, we are very interested because, as we already know, it's already happened with the pandemic, but we know specifically with antimicrobials that we are having a huge problem, that antimicrobial resistance, we have been talking about that. And the idea is that we need an increased need of new therapies. So what we decided to do is to work in 3 different ways. One way is we sequence, and it's not involved in now particularly in this project, but we sequence patients to understand their relation with the pathogens. But the other thing we do is to sequence the pathogens, then we go and analyze the whole context of that pathogen. Then we go and build protein structures. Then we go and analyze those protein structures and look for specific pockets. Then we go and try to find specific drugs that bind to those specific pockets. And that's what we have been doing in the last few years. For example, last year we published a paper where we identify a new target in tuberculosis, and we ended with a crystal structure with a drug, and we tested in clinical samples. So that's the whole idea. But each time what we are trying to do is to move the barrier that bioinformatics is doing a little more. So when we started here, we published this paper in 2018. We developed a whole pipeline to get what? Protein structures. And now we have AlphaFold. So what we just did is to take all the structure from AlphaFold and plug it now back to our pipeline. So the first thing is, the first paper that we published we had the structure of only 30% of many of these pathogens. And now we have 100% structures. So that's a huge change. And we are doing it really fast. We are able to put everything now all together. So what we do here is, so we get a sequence, we get different sequences from different strains, then we go everything into structure, then we analyze each of the properties of each of the genes. One interesting one is to use, for example, metabolic reconstruction of many of these pathogens. That is interesting because we want to analyze which are more important for each of the pathogens. Then, as I was telling you, we get all the information together in our pipeline, and we also add the protein structure and the pockets inside. When we calculate each of the pockets of each of these proteins, we get the drugability score. We develop a drugability score that allows us to tell us which pocket is more amenable to bind the drug. So we are adding again all this information to the same tool. At the end, what we have is different genomes. With these different genomes, they are all uploaded in our software. We are able to go and search for each of these pathogens and analyze them, not only in the context that we get from the bioinformatic tools, but also in the context that we get from specific experiments. So if you look here, I put collaboration open, and all these people are from Latin America. So these are all collaborations in Brazil, Uruguay, Peru. And what we do is we collaborate with people who already is working in one of these pathogens. And they want to integrate all the data and to be able to analyze the characteristics of their proteins and be able, if they are good, to identify new proteins and maybe use to develop new antibiotics. So in this sense, each time I go to a meeting, I find a new collaborator. So I hope we find new ones here. Once we get that, our idea here is it's not easy to understand what is a good protein to develop an antibiotic. It's not something, it's not the same question for each pathogen, because each time, imagine, we want to train an artificial intelligence algorithm that tells us which protein is the most important because we have a database. But the question is not easy to solve by artificial intelligence. So we are already working in changing this with ChatGPT 4.0. We already have a student trying to put all the information and be able to ask questions. But what we have right now is that on the fly, we use keywords, we select each protein based on, for example, it has a structure. Now all of them has. It has a drugability pocket. It's a choke point in the metabolic network. What about human off target? What about microbiome off target? We also look for that. But for example, you can upload in this database your own experiments. For example, you have overexpression experiments in certain conditions that is important for you, or you have experiments that represent the more expressed protein when you have a resistant strain. All that information goes together here, and we make a score that allows you to select the best proteins. Well, what we want to do in Cabana NET, we ended here in the last few years with each protein a pocket, and then we go and do molecular docking, bioinformatics. But what we are doing now is something a little different. With Cabana, we got a student to go to EMBL. And what we learn is all the Campbell database. Obviously, we learn to incorporate all the PDB database. So now what we have is for each of the pockets of each protein, we have the characteristics, but we have if a drug is bound there in, if it has been done in PDB, we already know it. If there is a similar domain that has a drug in that pocket, we also get that information. Also, if there is an assay that somebody made towards that gene, I said there is a binding drug towards that pocket. But in this case, you don't know exactly the pocket. You know the gene sometimes if you don't have the structure. So at the end, the idea is like this, and we have already done quite a bit, is you get all the proteins go to in our pipeline, and now you get for each pocket different drugs. So what are these drugs? Possible candidates to develop new drugs. Why? Because they have been identified in the same pocket, or they have been identifying a similar drug, or there is an assay that has already been done. And then what we also do is we have a database of all available compounds. The best one known is zinc, and we compare by drug similarity each of the drugs that are in this database with the drugs that appear here. So we are able to propose new candidates. And what we expect to have I think it's almost finished, is that you not only are able to look for proteins, you are able to look for drugs. So you are able to mix both things. So you have each drug, you get each gene, you get all the drugs associated to that gene, but then you can also look for drugs and go back and do. We have already done, for example, drug repositioning papers. So the good thing is with these kind of platforms, and especially focus on pathogens that we care in Latin America, we are able to accelerate this. For example, now we are collaborating with a global alliance in France that is, they have already identified some pathogens, and we are doing a joint effort to analyze each of the possible proteins, and we are offering them special leads to develop new drugs. So what our next steps with Cabana during this year and next year is, now we are integrated all the genomes. So each gene in all the genomes, we have drugs associated to. Then we would like to search by drug information. And the other thing that we are now going to do, and we are offering secondment positions right now, is to do two things. One is to develop artificial intelligence algorithms that allow us to tell which is the best drug for that pocket. And the second thing is to upload new pathogens. So we will probably establish a new collaboration to take a new pathogen in there and do the whole process, and then propose new proteins and new possible drugs. So thank you.
Okay.
Thank you, Loren. Very nice talk. When you were mentioning at some point microbiome of target effects, how are you, because you mentioned that some of the conditions is that and I'm saying because one of the big problems with antibiotics is that they attack the microbiome as well, and that it's a big problem of regular antibiotics. So you're thinking of new drugs, and but one of the conditions is that the checkpoint of the enzyme that you're attacking is central in the metabolism of that pathogen. But you don't want to have microbiome of target effects. I will guess that most of those central metabolisms are going to be shared between different microbes. So how can you get it to fulfill both conditions?
What we do is what we can do. So the first thing is to look. So we have the database of common microbes in the gut microbiome, and then we do blast to analyze if you have a cutting edge. So you say that gene is not present with similarity in many of these proteins. The second thing we do, and we are now able to do, is to analyze the pockets. So now we are comparing pockets. So you expect that the same protein in another bacteria, if you are using, has at least a different pocket. And we do that also for resistant mutations. So when we map in the whole pocket, we look that the resistant mutation, for example, are located in the pocket or are away. And that will explain some of that part. You are not able, you have to do the experiment if you really want to tell. But that's as far as we can go.
Okay, I'm sorry for the other questions, but we're slightly behind schedule. So I just want to catch up. And we have our last speaker of the day, another long-term colleague from the Cabana project and now Cabana NET, Andres Gatica from the University of Costa Rica. The subject is the genomic frontier, seeking climate adaptive vanilla varieties, utilizing cutting edge genomic tools. Andres holds a doctorate in agricultural science from the University of Hohenheim in Germany and is currently professor at the University of Costa Rica. He does a lot of mentoring for students and promoting bioinformatics in the region. In recognition of his accomplishments, the University of Costa Rica bestowed him the title of Humboldt Professor in 2023. So congratulations. Over to you.
Thank you very much, Ian. And thank you very much for inviting me to talk about a different topic. To close this session, you are not going to hear any more about human pathogens. I'm going to talk about famous and I think favorite spice, in this case, vanilla, and how to use genomic tools to improve this crop. First of all, I have to say that I am not a vanilla expert. I am the coordinator of this project, and this project is an effort of different institutions in Costa Rica, University of Costa Rica, and the Lancaster Botanic Garden. In Mexico, we have collaboration of Alfredo Herrera Estrella from Sinvestaff, and we have also collaboration from Belgium. Why vanilla? First of all, it's a high valuable cash crop for many smallholders in tropical and subtropical countries, including Costa Rica, and globally is a highly appreciated spice used in a variety of industries. Yesterday, we heard from Marco that is used to mix vanilla with coffee. Personally, I don't like it. But as many other crops, climate change is affecting the productivity of vanilla, and also the domestications of these crops reduce genetic variability, as we also heard previously in the case of coffee. And this reduced genetic variability may cause some problems related to biotic and abiotic stresses. So what is a solution? Since vanilla species are very close related to one of the most cultivated vanilla crop, which is vanilla paniflolia, we are interested in use crop improvements in order to give resistance against biotic and abiotic stresses, but it is not easy. In that case, if we have several vanilla species, we have to decide which species are we going to use to create these new improved varieties. And that's why our approach is to use advanced genetic tools in order to first to analyze the intra and interspecific diversity in order to confer resistance against climate change factors. In that sense, the main goal of this project is to use advanced genetic tools to identify resilient vanilla species in face of climate change. And we have two objectives. The first one is to create a precise and high quality reference genome assembly of vanilla paniflolia. Although there is a reference genome, it is not enough. It is not complete at all. And the second one is to evaluate genetic diversity present in cultivated as well as in wide relative vanilla using molecular markers. In order to do that, it looks simple, but it's not simple. We propose to collect samples in the field in Costa Rica, Mexico, and Colombia. Sampling has been complete in Costa Rica, and we have around 200 accessions. Then we develop an optimized protocol for DNA extraction. Several protocols were tested, commercial kits, and in-house protocols, because one of the main problems with vanilla is that there are a lot of secondary metabolites and that DNA quality was not good enough for sequencing. And then with the Cabana NET, we expect to sequence these accessions during January, April 2024. And we move finally with the help of one secondary, we expect to do the bioinformatic analysis. At the end of the project by October 2024, we expect to have a better reference genome of vanilla paniflolia in order to use this genome for SNP identifications. Also, to have a better description of genetic diversity in vanilla populations. One secondary trained and 3 scientific publications. So thank you very much. And I hope in the next Cabana meeting, I can present you some of the results.
Wow. So we've got time for a few questions. You only took 6 minutes. You had another 4.
It's because
Thank
I don't
you.
have any result to share.
Kat.
Thanks, Andres. We have an industry program at the EBI, and it works on a subscription basis. So mostly large multinational companies join the program, and we have knowledge exchange meetings, and this tends to drive projects that are of interest to industry. One of the partners is Unilever, and they have a huge interest in vanilla. They make a lot of ice cream. I think they've probably done their own sequencing and not released it. And I'm just wondering whether it would be worth us talking to the industry guys and perhaps inviting you over to one of our quarterly meetings to give a talk to see whether we could explore some synergies there. I'd actually like to think more broadly in terms of, all of the crop related talks that we've heard today, and maybe we do something that's a little bit more expansive, but I know for sure that there's a lot of interest in vanilla.
Thank you very much. We appreciate very much if you can give us the contact, because previously we were discussing with Alfredo that in the second part of this project, we would like to sequence other species of vanilla like Vanilla pompona and Vanilla odorata. And for that, of course, we need a lot of money because we want to do the whole genome sequencing with a good coverage.
Hi, Andres. Thank you. I'm sorry if I missed that, but are you planning on using the same rat sequencing approach that you use for coffee for this project?
No, we at the beginning of the project, we think about that to use DD rat. But now, since we have a collaboration with Belgium, we are going to use dark sequencing.